Good morning and welcome to today's Plant Treasures webinar. We look forward to discussing the role of the nursery and living collections in conserving native plant species. My name is Amelia Martyn Yenson and I'm a project manager at the Australian Network for Plant Conservation. In the spirit of reconciliation, the Australian Network for Plant Conservation acknowledges the traditional custodians of country throughout Australia and their connections to land, sea and community. We pay our respect to their elders past and present and extend that respect to all Aboriginal and Torres Strait Islanders and all First Nations peoples joining us today. Please use the chat button at the bottom of the screen to introduce yourself, let us know where you're from, and if you're in Australia, you might share the names of the traditional owners of the lands where you are today. Our discussions are based on the new third edition of Plant Germplasm Conservation in Australia, funded by the Ian Potter Foundation. You may also know this book as the Germplasm Guidelines. In this edition, we have a new chapter on the role of the nursery in ex situ conservation and a fully revised chapter on living collections. It's a great resource and available for free download, and you'll hear more about the guidelines shortly. Today's webinar is a special event planned to coincide with Botanic Gardens Month, and we welcome all ANPC and BGANZ members joining us today. One thing we all have in common is our passion for plants, and if you're not already part of these conservation and botanic gardens networks, we'll let you know how you can get involved during the day. I'd like to thank the ANPC staff, Jo Lynch and Chris Fernance, and the BGANZ Council and their Communications Officer, Dr. Rebecca Harcourt, for their assistance in today's webinar. Botanic gardens play a great role in conserving and protecting the flora, and through that, the fauna of Australia and New Zealand. We're thrilled to be introducing you to experts from botanic gardens and conservation organisations around Australia today, so they can share their insights into current practices and future needs of this special part of the horticultural industry. There'll be plenty of time for questions for our presenters, and we'll conclude the day with a panel session. Before we start the day, I have a few questions for you, our audience members. So during the webinar, we'll invite you to participate in a Mentimeter poll. For those that haven't used Menti before, it's very easy to use. The instructions are on the screen and we will step you through using it. There we go. We're hoping the answers to the surveys today will help us understand who we are reaching through our webinars, the unique skill set and experiences of people in this sector, and how we can improve our collaboration in future to better meet the conservation needs of Australia's amazing plants. So we'll start with a quick poll to get warmed up. If you haven't used Menti before, the instructions are on your screen. You can respond via the Menti app or by opening a web browser and entering the code on your screen there. Or you can hold up your phone and use the QR code. I'll just wait a moment for people to join the poll. That's fantastic. So the Mentimeter code for this poll is 23200967. Fantastic. And those instructions will stay up on the screen as we go through. First of all, we'd like to know where you're from. Excellent, we can see people voting. We've got a good spread across the East Coast. We've got people joining us from New Zealand and other places outside Australia. Welcome. Western Australia, Queensland. Christine is in Tassie, she's our ANPC Communications Manager. Wonderful, fantastic everyone. We'll just wait a moment in case there's other people ready to vote. I love seeing all the dots race across the screen. Fantastic. Thanks so much for joining us today. And now we'd like to know where you found out about today's webinar. And you might have heard about it from more than one place, so you can choose more than one option here. Hopefully you've seen it advertised in several places. And we'll give you details later on how you can subscribe to both the ANPC e-newsletter and the BGANZ newsletter, which are great resources to hear about events like today's and the Botanic Gardens Month events. Fantastic. And thanks for everyone who let their colleagues know about today. We really appreciate that support as well. People encouraging each other to tune in. Fantastic. Thank you. And finally, we'd like to know if you're using resources like the Germplasm Guidelines, the Flora Bank Guidelines, or the Translocation Guidelines in your work at the moment. And we've got options here, whether you're using the new current versions that have come out or the previous edition. If you haven't had a chance to look at these guidelines yet, we'll be providing links throughout the webinar today. And we've also created a web page to help you select the right set of guidelines for your situation. So that can be accessed via the ANPC publications page. Fantastic, and lots of people already using the guidelines. The Flora Bank Guidelines for ecological restoration that has a lot of information about seeds, which is excellent, and the Translocation Guidelines, which have been out a few years now. Fantastic. If you haven't heard of the guidelines before, welcome. I'll give you a quick overview of the content in the Germplasm Guidelines shortly, and absolutely acknowledging that there are other resources that are more relevant to people depending on their work situation. Fantastic. Thanks so much for that. Okay. I'd now like to invite John Arnott, the BGANZ Council representative for Victoria, to introduce today's theme. John has held the role of Manager of Horticulture at the Royal Botanic Gardens Victoria, Cranbourne Gardens since 2008. He's provided inspiration and leadership to their growing team of horticultural staff, with a key focus on the horticultural management of the Australian Botanic Garden and facilitation of a range of conservation horticulture activities. Prior to this, he worked in significant public gardens, including Melbourne Zoo for over 20 years and the Geelong Botanic Gardens. In 2015, John was awarded the Australian Plants Award by the Australian Native Plant Societies to recognise his outstanding contribution to knowledge of Australian plants. We're so pleased he could join us today. Welcome, John.
Thanks so much, Amelia. I am joining the meeting today from Bunurong land and in Bayside, Melbourne. Look, thanks so much for the opportunity to be able to introduce today's theme. This whole conservation, horticulture, ex situ conservation, the role of living collections in supporting plant conservation outcomes has been something that it's been a focus of mine for a good many years. Indeed, it's a passion, occasionally bordering on an obsession. So it's actually lovely to be able to join today's meeting and do a quick introduction. I'll probably try and wear a few hats by way of introducing the theme and reflecting a little bit on BGANZ, ANPC, and the sort of the evolution of conservation horticulture in botanic gardens. So, first hat I'll pop on is a BGANZ hat. So BGANZ is excuse me, Botanic Gardens of Australia and New Zealand, and it is lovely. I agree, Amelia, that this collaboration between BGANZ and ANPC is it's to be applauded, and it's something that we should be doing potentially a fair bit more of into the future, because both BGANZ and ANPC are very strongly aligned. BGANZ really started in Canberra. It came out of essentially came out of the Council of Heads of Botanic Gardens, where all the major capital city gardens, they wanted to pose the question to the Botanic Gardens community in Australia in the first instance, whether there was any interest in establishing a sort of a national network of Botanic Gardens. And there was a congress held in 2001, where over 100 delegates attended that Congress. The main question that was being asked of the Congress is there an appetite, is there a need? Would the Botanic Garden Network benefit from establishing the Botanic Garden community benefit from establishing a network of gardens? And the resounding resolution from the meeting was yes. So, under the guidance of Phil Moores, who was Melbourne Gardens, the Royal Botanic Gardens Melbourne Director and Chief Executive, he was the initial chair of BGANZ and wrote up some terms of references. It became really obvious that New Zealand should be incorporated into this network. and there was an inaugural meeting of the group at the Geelong Botanic Gardens in 2002. I just started at the Geelong Botanic Gardens at that point, and found myself on the program committee and almost project managing the Congress. It was an incredibly successful Congress. It attracted 300 delegates from all around the globe. BGANZ was sort of off and running, and me as an individual, just by luck, happened to be sort of right there in the thick of the establishment of this emerging network. The ongoing success of BGANZ, it's really, again, parallel with the ANPC, is the focus of the organisation in supporting regional botanic gardens and supporting its membership, building capacity within its membership to actually fulfil the roles of a botanic garden. Look, the majority of botanic gardens in Australia and New Zealand are funded by local government. Local councils are concerned with all matters, of things close to our home. Building regulations, public health, roads, footpaths, playing parks, fields, rubbish, libraries, local environmental issues, a whole range of community services. And botanic gardens are competing in that space. We recognise that there's a distinction between a botanic garden and a high quality public park. Superficially, you'd look at something like Rona Street in Queensland, or Fitzroy Gardens or Royal Park, Centennial Park Gardens, and they look similar to botanic gardens, but there's this distinction between a high quality public park and a botanic garden, and that's this curated collections. The distinction being that botanic gardens are about living collections that are curated for specific purposes, how they're acquired, developed, maintained, documented, interpreted, and displayed. There's some real specific aims that sit behind the curated collections of collections in botanic gardens, that the distinction to local parks. So back to that local government, regional botanic gardens are absolutely funded by local government authorities, but given the amount of competing resources and staffing, community consultation, liaison, resourcing, programming, infrastructure maintenance, but it's really unlikely that if you're a curator at a botanic garden with 1 or 2 staff, that you could go to your parks management, or you could go to your council and look for nuanced technical support around living collections management. And that's where sort of BGANZ comes in terms of building capacity for regional botanic gardens in Australia and New Zealand to be botanic gardens. And that's fulfilled for a whole range of toolkits. There's journals, congresses, regular network meetings, technical working groups. There's a collections working group and an education working group. And we're currently working on a database project that we're looking to roll out to members. So, building that capacity is a really important element of BGANZ as an entity. BGANZ has just changed governance. We've gone from an incorporated business to a company limited by guarantee, and I think the next iteration and evolution of BGANZ is about to happen. I will, if I was clever enough, I'd put a couple of things into the chat, but I'll do this after this. It's Botanic Gardens Month, and there are a host of activities. I'll drop some links into the chat, but particularly there's a thing called Plant Passions, and we're asking folks in the plant world to submit a 30-second video or a minute video on a plant that they that they're keen on, or a plant story, or just a series of plant story videos from people that are into plants. So I'll drop a little note into the chat. With regards to ANPC, there's a link here, because ANPC was also inaugurally formed at the National Botanic Gardens at Canberra. In 1991, ANPC was formed really very strongly as a botanic garden ex situ. It had a very strong ex situ plant conservation focus. And the inaugural congress or conference of the ANPC was entitled Protective Custody, and it was really looking at ex situ plant conservation in Australia, looking at similar objectives to BGANZ, and really this is interesting. This is 1991. It's a good few years ago now. So the initial driver for the ANPC was, coordinating ex situ plant conservation activities. And, the ANPC has had this remarkable evolution from that really, really strong focus on ex situ conservation. And it kind of morphed to more encompassing integrated plant conservation, using a whole range of tools and techniques for conservation outcomes and, on-ground, in situ plant conservation, plant management, translocation, managing threatened processes in the wild. The number of integrated plant conservation disciplines sort of merged from something which was very, very strongly ex situ, and ANPC really diversified, from that ex situ conservation to be much more holistic in terms of conservation outcomes, and using a range of tools and techniques. And that's, reflected in, the conference themes over the years. So ex situ conservation always has and always will be an absolutely key focus of the ANPC, and that's reflected in this magnificent recent review of the Germplasm Guidelines, which forms the basis of much of today's content. So ANPC, BGANZ, very complementary organisations, and long may this association continue to thrive. And thanks again for the opportunity for BGANZ and for ANPC to come together. But today's really about conservation horticulture, so I'll put on my horticulturist hat. And again, just reflecting on horticulture across the career, it's really evolved from. It was originally, when I first started in horticulture, it was called amenity horticulture, quite literally focused on improving the visual amenity of public places through activities such as spring and autumn bedding and ornamental display. Horticulture was about ornamentation. Horticulture was about beautification. There's been these significant trends that have taken horticulture from amenity to much more focused pursuits. The emergence of the indigenous plant movement, the indigiphiles, and the local provenance grow plants movement, which hit great heights. Environmental horticulture emerged with a focus on sustainability, water management, and those types of things. There's more recently the Gardens for Wildlife movement, managing urban biodiversity and ecology, and again, necessarily preparing for climate change and climate adaptation, building resilience in urban planting. So there's been this incredible evolution over the last 30 or 40 years, from ornamentation to horticulture responding to a sort of this rising tide of environmental awareness. Botanic gardens were absolutely involved in these activities, and sometimes were leading those agendas. But I guess the point of difference for botanic gardens is this term conservation horticulture. And conservation horticulture it's emerging. It's always been there. 1997, ANPC had the list of activities which are associated with ANPC that was the inventory of conservation horticulture techniques. So it's been around for a little while. But potentially there's been a few impediments to botanic gardens picking up conservation horticulture. Many botanical institutions, conservation has remained in the domain of scientists, and potentially horticulture has been overlooked as a key component of conservation projects. Conservation has been the remit of field botanists, conservation botanists, conservation geneticists, mycologists, ecologists, pathologists, taxonomists. But gee whiz, when you add horticulturists into that field, it actually provides incredible opportunities to enhance the work of those plant scientists that we just listed before. And that's because as horticulturists, we grow plants. We're really good at growing plants. We can grow a huge range of plants. We can collect plants. We can document those collections. We can propagate plants, bring things into cultivation for the very first time. work out propagation cultural protocols for plants that have never been grown before. We can hold plants in living collections for generations for all manner of activities, monitor plant performance in cultivation, understand environmental tolerances of plants. We can talk about plants until the cows come home. We can distribute plants from garden to garden and establish multi-site holdings. We document our living collections. We can share our data with other gardens and conservation agencies. And they're just some of the services that horticulture can offer plant sciences. That conservation horticulture agenda is really what we're talking about today. And today we're going to hear from a range of a beautifully curated program, I must say, Amelia. We're going to hear from a range of conservation horticulture practitioners. I, for myself, are really looking forward to hearing from the folks today. And thanks for the opportunity to just reflect a little bit, Amelia, on conservation horticulture from an idea in 1997 to something that we're practicing today and hopefully contributing in a really positive way to conservation outcomes through doing what we do well, growing plants.
Fantastic. Thanks so much, John. And what a great way to start the day with reflections on the beginning of BGANZ and AMPC, conservation horticulture, and the role of botanic gardens now and in the future, and how integral our horticultural expertise and staff are in that area. We now have a video to introduce today's theme, and you'll hear what John and also Warren Worboys talk about living collections and the role of the nursery at the Royal Botanic Gardens Victoria, Cranbourne Gardens. Warren Worboys is the Curator of Horticulture at the Royal Botanic Gardens Victoria, Cranbourne Gardens. He's worked in a diversity of roles managing the nurseries, garden development, and also looking after Indigenous bushland at the Melbourne and Cranbourne Gardens. He's travelled extensively across Australia, studying the flora and collecting and propagating plants to extend the RBG Victoria's collections of wild provenance plant species. Enjoy the video.
When people think of living plant collections, the image of plants in pots often springs to mind. But living collections are more than just nurseries.
So there's living collection holdings, and that's holding threatened species as living collections in gardens or in pots. There's holding material for research purposes and, understanding the environmental tolerances of a species. There's growing plants with the view to put them back into the wild, so recovery programs. There's a really important role for public gardens and botanic gardens to actually tell conservation stories through living collections.
These living collections differ from standard nurseries or even collections used for restoration because individual plants are often tracked to maintain their unique genotype and ensure that genetic diversity isn't lost. Plants in living collections are propagated in three main ways: seed, vegetative, or specialist methods like micropropagation or tissue culture. Storage and propagation from seed is cheaper and requires less space, but there are limitations. It takes longer, especially if dormancy needs to be alleviated. You don't know the exact genetics of the plants, and some species produce limited seed, no viable seed, or even no seed at all. Vegetative reproduction then becomes a cheap and often reliable method to produce large numbers of plants.
It could be growing from cuttings, grafting, layering, so there's aerial layering, below ground layering, division, and that could be from dividing rhizomes, etc. It could be stem cuttings or stolen cuttings from rhizomatous plant material.
Vegetative propagation relies on totipotency, the ability of the plant to regenerate using unspecialized cells in roots, shoots, rhizomes, and leaves. Vegetative propagation produces clones, which is a good way to maintain a nursery population that genetically represents the wild populations.
Cultures are hardened off, and then grown on into containers and reintroduced into the nursery.
But with threatened species, nothing is straightforward, and often different propagation methods are trialled. One of the most common and often first tried is semi-hardwood cuttings collected in spring or autumn. A tutorial on this method is included as a separate video in the series. Propagation and maintenance of threatened plants in the nursery also offers a rare chance to learn about the biology of a species, including which ones have specific mycorrhizal relationships or which require extreme growing conditions like acidity, waterlogging, or mimicking high humidity, such as occurs in a cloud forest.
So the collection behind me here is a collection of the endemic taxa from 8 or 9 far north Queensland peaks. These peaks, they're cloud forests, and all the climate modelling is suggesting that these peaks will get hotter and drier. Yet there's 70, 80 endemic species that only occur on these peaks. They occur nowhere else on planet Earth. So what we've established here as a part of this consortium is an ex situ conservation collection.
Learning how to propagate species in this collection has taught the team about plant growing conditions, and in some cases, they've been surprised at unexpected tolerances.
The assumption is that if it comes from a narrow ecological niche, it might have a narrow set of environmental tolerances. But we're finding out with a couple of examples, there's a Primnopotes, which we've been cultivating in the garden for a good number of years. It's tolerating 40 degrees and low relative humidity. Yet it's one of these things that come from the mountain peaks. So its environmental tolerances are much, much wider than what you might think.
Some threatened plants are extremely susceptible to disease, and living collections give a great controlled opportunity to test potential treatments. But living collections do have downsides.
The amount of record keeping, the human resources required for actually conserving the material, etc., are really quite high. So it can be problematic to actually get sufficient funding and the resources to actually manage a collection.
That is why collaborations are becoming more important.
The aim for this collection is to not hold it just here, it's to hold it here and in a good number of other botanic gardens up and down the eastern seaboard. And that's referred to as a meta collection, so multi-site conservation collection, which is bringing in a number of different agencies. The idea there is that if a particular plant in our collection here died, and that's what happens in living collections, there's a sort of a backstop population. So yeah, having conservation holdings not just held in one location is a really legitimate thing, and it's starting to happen a lot more in botanic garden worlds.
There is no simple single method for best practice ex situ conservation. The nursery plays a key role in getting plant species into storage, then at the right time, getting them out of storage and regenerating germplasm into healthy living plants. It takes a suite of specialist skills and resources to conserve our unique threatened flora. More information about ex situ conservation methods is included in Chapter 8 of the Guidelines. Chapter 11 also covers all the different types of living collections you may encounter to conserve our national plant treasures.
Thanks so much, John and Warren, for a great introduction to the topic in that video. I'd like to thank Chantel Doyle and Michael Lawrence Taylor for their work on this video series. These videos and the webinar recording will be available on our ANPC YouTube channel in the next week or so. You're welcome to put questions for John in the question and answer box that you can see on your screen so that we can ask these shortly. I will note that some of the people appearing in the videos today are not appearing live, but their colleagues are on screen can definitely answer some questions for them. I'd now like to give a more detailed overview of the Germplasm Guidelines so you know what kind of information is available. And I'll also take a look at how ex situ conservation supports in situ conservation, which John mentioned in his intro, and it's also a theme that will come through different presentations today. Now, bear with me while I get my presentation sorted. Okay, fantastic. So I'm going to give a quick overview of the Germplasm Guidelines today. And before we begin, I'd like to acknowledge the traditional owners of the lands where I live and work, the Darug and the Darawal people, and I pay my respects to the elders past, present and emerging. This is the third edition of the guidelines for ex situ plant conservation. So as you've gathered, that's plant collections held off-site or away from their natural habitat. They were updated by 78 contributors with oversight from the Germplasm Guidelines Steering Committee, and the project is funded by the Ian Potter Foundation. The guidelines are available for free download, or you can order a print copy. It's a great resource for the nursery, for your library or your tea room. Germplasm is a term for any type of plant tissue that can be turned into new plants, including seeds, cuttings, spores and pollen. In the guidelines, we have chapters on seed banking, tissue culture, cryopreservation and living collections, which are all germplasm collections that make it possible to capture genetic diversity for the future. An ex situ conservation strategy should aim to capture genetic variation at the species, population, individual and allelic levels, as this level of diversity provides the basis for adaptation now and into the future. Ex situ germplasm conservation enables use of this captured diversity in a variety of ways, primarily to enhance in situ conservation efforts. If we go around Australia and look at the germplasm collections of the Australian Seed Bank Partnership and its associates, you can see that seed banks and conservation nurseries are the most common type of germplasm collection. Seed banks and nurseries can be found at many state-based botanic gardens, as well as within other organisations like the Australian Grains Gene Bank and the Australian Pastures Gene Bank that store wild relatives of crop species. So this map from chapter 2 of the guidelines doesn't include the smaller restoration seed banks that store wild collected seed or the regional botanic gardens that grow Australian native plants. So this is not an exhaustive list, but it does give you some idea of where you can go for advice or assistance. You can see again the range of ex situ collection types in this image in the grey box. You've got cryo storage, seed banking, micropropagation or tissue culture, and living collections. The capture of genetic variation, the scale of the seed or cutting collection, and the time frame for curating ex situ collections depends on the project goals. These ex situ collections provide both information and propagules to support in situ or on-site conservation, and they rely on techniques such as seed germination or nursery propagation. You can see the management activities for in situ populations, so the natural habitat, in the green box there. Depending on the site conditions, the management actions include maintaining habitat in a viable state, promoting natural regeneration, assisting regeneration, and more intensive activities such as direct seeding, planting, and translocation. And some botanic gardens carry out restoration-focused activities, including monitoring and management of threatened species. As John noted, the specialist skills and expertise within the botanic garden sector is so important to supporting both ex situ and in situ activities, and we'll return to this image later in our panel session. The guidelines begin with a chapter outlining the ex situ options available, what you need to consider, and how to best prepare for germplasm conservation. Throughout the guidelines, we've included graphics like this, such as flowcharts, pop-out boxes, and references to source documents. So it brings together lots of information in a way that's easy to use. It's a really good reference for things like license considerations, how to best capture genetic diversity, seed collecting and propagation tips, and processes for each type of germplasm collection. You can dip into the sections you need or use them to find out perhaps who else in Australia is working on a particular technique or a plant family. And you can do that through the chapter author lists and the references in each chapter. If you take a look at this flowchart, and you don't need to see all the detail, I know it's very small, you can notice that there are a lot of things to consider in the pink and grey boxes at the top, but before you even start making a seed or cutting collection for conservation purposes. If you're new to the process, talking it through with an experienced staff member at a botanic garden is a great way to get up to speed. These people may be based in the horticulture team, the nursery, the seed bank, or you may need to speak to a few people to get the appropriate combination of germplasm for your target species. We now zoom into the bottom half of this graphic. So you've had a chat to someone who's experienced in the process, collected information about the target species, and considered how the collections will be used, which could be years or even decades after collection. So then we can go ahead and collect seed or cuttings or other material. Collections are then processed and stored ex situ using one of the methods you can see on screen with these neat icons. First, to note that we have a table in the guidelines that gives an overview of the advantages and disadvantages of each germplasm collection type. In most cases, seeds are the lowest cost and easiest to maintain, taking up very little space. Although many Australian species have very long-lived seeds, we need to remember that seeds don't last forever, so we have to have a way to use them and regenerate or recollect that species in the future. The other methods offer particular advantages, but require a higher level of technical skill to get into storage. So it can be time-consuming and expensive to develop protocols for each species. They also require more maintenance, which can really multiply labour and resource costs in the long term. The guidelines include updated chapters on seed collection, banking of orthodox seeds, germination, and dormancy. They have practical information that can be really useful for testing seed quality and planning germination experiments. We also have a new chapter on identifying and conserving non-orthodox seeds, which can't be dried or frozen, or maybe can't be stored long term. Sometimes we call these exceptional species. So it includes species where seeds are not available, species, as I mentioned, with desiccation sensitive, freezing sensitive or short-lived seeds, and also species with deeply dormant seeds. Often these species need to be conserved as living collections or using a combination of tissue culture and cryostorage. If this interests you, there's plenty of additional information available, including videos on our ANPC YouTube channel. In this edition, as I mentioned, we have a new chapter dedicated to the role of the nursery, because most collections pass through the nursery at some stage between collection and use. For example, seeds germinated at the seed bank may need to be grown on and distributed. So we have lots of information about vegetative propagation, including cuttings, division and layering, grafting, and budding. Vegetative propagation is often used when seed is not available, of poor quality, or can't be germinated easily. It's also used when individual plants need to be replicated for conservation. In this chapter, we have case studies on propagation techniques such as rhizome cuttings and use of cellular plugs to improve cutting survival rates. Record keeping is particularly important, so the guidelines include some tips on maintaining good records for the lifespan of the collection. And we suggest you take a look at our previous webinar on record keeping and the BGANZ resources available on this topic as well. This chapter includes case studies on planning and partnerships, so you can find out more about the meta collections of threatened species from the cloud forests of Far North Queensland, mentioned by John. We also include the work of the South East New South Wales Bioregion Group, conserving scrub turpentine, Rhodamnia rubescens, in collaboration with regional, state and national botanic gardens. We also get an insight into the use of genetics to inform the conservation strategy for the Nerriga grevillea, Grevillea renwickiana. As you heard from John and Warren this morning, living collections include botanic gardens and arboreta, field gene banks, seed production areas or seed orchards, and nursery collections. These plants can provide material for restoration, translocation or research, education, horticultural development and display, depending on how the collections were made at the time. For key species, meta collections provide a duplicate or backup at other locations in a similar way to the backup of the ASBP seed collections at the Millennium Seed Bank, for instance, or the agricultural seed collections at the Svalbard Global Seed Vault. Living collections are important resources, particularly when access to plants in situ is difficult or further endangers the species. So the Wallamai pine is a good example here, with more than 100 papers published based on work on ex situ collections. The emphasis in this chapter is that the purpose and the number of plants in living collections needs to be considered at the time of establishment and throughout its maintenance, so that we understand the genetic diversity that we have captured and available for use. To illustrate this point, we have a photo of botanist Trevor Wilson and conservation geneticist Samantha Yap with Amelia spicata, a threatened species, at Mount Annan, where they helped work out how to best capture genetic diversity while maintaining an appropriate number of plants. You can see some images from the case studies within this chapter here. So we have studies on species such as King's Lomatia from Tasmania, the native guava from New South Wales, and rare and threatened species in a Care for the Rare program in Victoria. We get an insight into the development of conservation collections and the orchid germplasm collection at the Royal Botanic Gardens, Victoria. And we find out about the challenges of bulking seed collections for seed restoration or conservation, which Lorraine will talk about shortly. We also have a case study from Auckland Botanic Gardens on ex situ management of plants that are highly susceptible to myrtle rust. In this edition, we've added a separate chapter on orchids and mycorrhizal fungi, the symbiont which needs to be isolated and conserved alongside the orchid seed. We also touched briefly on the isolation and storage of rhizobia, which are symbionts on the pea family, Fabaceae. In this edition, we also have a new chapter on special collections. So in this, we've included taxa that are often underrepresented in ex situ collections, or they have special growing requirements. So things like ferns, carnivorous and parasitic plants. Our understanding of ex situ storage requirements for these species is still developing, and it's an area that's ripe for research in the future. The addition of these two chapters and more information throughout all the guidelines chapters means we're capturing information on effective ex situ conservation for the widest possible range of species. So we can build on the experience of others who are dealing with similar taxa and germplasm types rather than trying to start from scratch each time. I'd like to acknowledge all the authors who contributed to the guidelines and particularly thank the chapter lead authors and steering committee members for their hard work and support. Please download the guidelines and share them among your colleagues and friends of the gardens and volunteers. And now back to the main program and our next video. This new edition of the guidelines includes lots of detail on propagation methods within chapter 8, as I mentioned. We now have a short video of Mandy Thomson from the Royal Botanic Gardens Victoria Cranbourne Gardens, where she explains the process of preparing semi-hardwood cuttings in the conservation nursery. Hygiene and careful plant handling are emphasised to maximise success and disease-free plants. Thanks so much, Mandy.
Vegetative propagation can occur in numerous ways, which are outlined in the Germplasm Guidelines. One of the most common is semi-hardwood cuttings of current or last season's growth. In this video, Mandy Thomson from the Royal Botanic Gardens Victoria, Cranbourne, explains the method.
So we enact the hygiene protocols. So we do a physical inspection of the pest for pest and disease. We'll drench in detergent and also in eucalyptus disinfectant with water and rinse clean before we start working with the material. And so clean methylated spirits and sharp secateurs and a sterile environment wherever possible. Sometimes in the field that's not possible, but in the nursery it is. And we just process that cutting material down into a cutting that will fit into this preformer plug, which creates 100% capillary action from the moment the cutting goes in. We generally wound the cutting just below a node where it's going to grow, and then we'll process that cutting material down just to reduce the transpiration that we might get. So we'll use just a hormone like this Clonex purple, and that's indole butyric acid, and it's just a bit of a dip in there, and then place that into the plug. We use the plugs because it reduces transplant shock as well as keeping 100% capillary action and keeping them moist. So you can imagine when that's struck and it goes into the tube, into a completely different media, a completely different environment, that you can reduce one of those factors by keeping it snug in its plug still, so you're not disturbing the roots too much. So that's the reason why we use these. They're not for everything, they're not for a lot of desert flora, but most of the material we grow here at the gardens can get through really well in a plug.
Once the cuttings have been processed, they are maintained in the nursery glasshouse until roots develop. Once roots have developed, cuttings can be transferred into forestry tubes with pasteurised potting mix appropriate to the species and the final planting location. For more information on this process as well as other propagation techniques, please see Chapter 8 of the Germplasm Guidelines.
Thanks so much, Mandy. That was excellent run through of that technique. Please keep questions for John and myself about the germplasm guidelines generally coming in the Q&A box, and we'll spend some time on these questions now. John, if you'd like to turn your camera on. I have some questions myself about your fascinating work there. I'll kick it off and we'll keep an eye on questions coming in from the audience today. So you mentioned the development of these meta collections for some species from the tropical mountaintops, the Far North Queensland project. Can you tell us a bit more about this? And how are these types of collections helping protect species that are at such risk from climate change and maybe disease incursion as well?
Sure. I think the project is called TROMPS. So that's Tropical Mountain Plant Species Project, and it is very much a consortium of groups led by James Cook University. So in the first instance, it's the tropical herbarium in James Cook University. So the project arose from academics and botanists in that part of the country, recognising that these threatening processes are in place for this unique flora. This flora with high levels of endemism. And at that point, with some funding from the Potter Foundation, which is relevant to this conversation as well, given the support that Ian Potter has placed on the Germplasm Guideline updates, there was a group of people, a consortium of people, including Botanic Gardens, who basically came together to scope the project. So it's one of those nice examples of collaboration, multidisciplinary participants. So plant growers, plant ecologists, plant geneticists, people that are looking to the potential for storing these things inside. The Native Rhododendron Society were in because it's the sites of the only known sites of Australia's two, let's call it two, species of rhododendron. So just inherently multidisciplinary. It's a funny one, this one. The ex situ thing feels a little bit like captive breeding in a zoo, which is kind of a bit challenging just in its own right, because if we're holding these things in ex situ holdings with little or minimal capacity to be able to put them back into habitat, it's bordering on a little bit of an ethical dilemma, this one, in as much as ex situ conservation has always and should always be viewed as a means to an end rather than an end in itself.
Yes.
But the potential for these in the very worst scenarios is that these things are held in living collections, and not able to be reintroduced into the wild. It's an interesting thing, but that's where
Yeah.
understanding the environmental tolerances come in.
Yeah, well, that's what I was going to ask you about as well. So you talked about some really surprising responses to growing plants in these different conditions, and I think the Promnoptes was one that you mentioned. So can you talk us through your approach to understanding the environmental tolerance? Is it quite structured, or is it a matter of trial and error based on your horticultural experience and your kind of knowledge of the ecology of the plants?
Look, ideally, it's not anecdotal, but a lot of it is.
Yeah.
But that's where databases and documentation and making those observations becomes a really critical thing for this project. And because the project is growing plants from so far outside their natural range, a big component of the project is doing that documentation and making those observations. Again, cultivating these things across a number of different gardens. So we're talking gardens in Tassie, gardens in Victoria, the Dandenong Ranges up in the mountains, some of the cool climate gardens. It's interesting that the Cairns Botanic Garden are not really able to participate in this because the physical conditions that they have in lowland tropical Queensland are just not suited to these plants. So yeah, I guess that network of growers who are making observations and documenting and sharing that information is going to build to the understanding of environmental tolerances of the species. The challenge is documenting that properly and then being able to communicate that. So databases, that's where the databases come in.
Being able to share the information. Yeah, often when it's the first time some of these species have come into cultivation, as you said, learning as much as we can about them. I noticed Warren, thanks Warren, he's just put a note in the chat of the 70 taxa considered under threat from climate change impacts on the far north Queensland high peak flora. 7 of those were evaluated to likely become totally extinct by 2050. That's pretty sobering, isn't it?
Yeah, it's pretty sobering. Yeah, so and that's where that plant zoo captive breeding dilemma actually comes into play. It's I've pondered that one a little bit, just philosophically, and just like I think it's the right thing to do to hold them. But
yes,
ex situ conservation it's not an end in itself, I don't think.
That's right. When we're always so focused on the project goals, what are we going to do with these species? Yeah. Thanks so much, John. We'll move on with the day's program to keep it to time. Really appreciate your input and to you and Warren for that fabulous video. So we're going to hear from staff at 2 botanic gardens now about their role in plant conservation and how these insurance collections help threatened plant species. And first up, I'm delighted to introduce Lorraine Perrins. Lorraine is the Curator of Conservation Collections and Subantarctic Flora, how fascinating, at the Royal Tasmanian Botanical Gardens. She oversees the ex situ potted collections and plant propagation for their collections, as well as supporting the Tasmanian Seed Conservation Centre by managing the seed orchards for the program. Lorraine also coordinates regular meetings of the Threatened Species Networking Group to encourage regular communication, information exchange and engagement regarding threatened flora in Tasmania. Lorraine's a regular contributor to the ANPC Bulletin, Australasian Plant Conservation, and today she expands on her most recent article, which addresses the trials, tribulations and triumphs in managing a botanic garden seed orchard. Thanks so much, Lorraine.
Good morning, everyone. My name's Lorraine Perrins. I'm from the Royal Tasmanian Botanical Gardens. I'd like to thank the ANPC for inviting me to give this short presentation to you today. Since the establishment of the Tasmanian Seed Conservation Centre in 2006, the Botanical Gardens Nursery has been actively involved in assisting the seed bank in achieving its goals of securing Tasmania's flora. The ways in which we've done this have been through our seed orcharding program. Seed orchards, or as they're referred to in the Germplasm Guidelines, field gene banks, are essentially pots of plants or plants in the ground for the sole purpose of harvesting a genetically diverse large collection of seed from. Why do we need them? Well, most of you who would do a lot of field collection work would know that field work can be quite challenging. Some species will only grow in really remote or inaccessible areas, meaning that to monitor those species you need to travel long distances, either by maybe boat or even helicopter. That's very time consuming and it's also very expensive. So to have those plants accessible in the nursery is a lot cheaper. Some species occur in small populations, but across large areas, meaning that accessing them can also be difficult at the right time, getting the timing right to harvest the seed. Some species continually flower and set seed over long periods of time, meaning that if you have to keep continually going back and forth into the field, that is also time consuming as well. So what are some of the things to consider when setting up your seed orchard? Well, apart from trying to get a lot of seed, the other thing that you need to consider is that seed needs to be genetically representative of the species that you're trying to harvest. The recommendations in the guidelines are that you will need about 50 individuals or different genotypes of each species to get that broad cross-section of genetic diversity. Now this can actually take a bit of time, and I can give you an example that we had. This little plant up here is Wormbia latifolia subspecies Vanessae, and it grows in northwest Tasmania. We were given seeds of this many years ago, grew them on. They're a little bulb. Bulbs take some time, as many of you would know, to flower. From the 18 plants that we grew, once they started flowering, we realized that only 6 out of the 18 were female plants. So this species is dioecious, needing male and female plants, individuals. And so we need to work on building up that population further to get more females into our orchard. But it has taken a long time before we realized this. The other thing you need to think about is understanding the biology of the species. And we'll talk a little bit more on this in the next few slides. But knowing what the pollinators are, what the seed dispersal mechanisms are. Also working out growing requirements of some of these species can take time too. As many of the horticulturalists listening today would know, a lot of these species that we grow in botanic gardens haven't been grown anywhere else before, and so you're starting from scratch and trying to learn the little tricks of the trade and what the plants need to keep them happy, and that can take time too. And the other thing is that you need to work out the most effective way to harvest the seed. How does the seed disperse? Does it just drop? Does it fly away? Do you need to bag it? Do you need to hand pick it? All those sorts of things need to be considered. Some other things to consider: are there any species growing nearby your seed orchards that might actually cross-pollinate and cross-contaminate your eventual seed collection? We had an example here. This lovely plant, many of you would recognize. It's Hardenbergia violacea. Grows along the east coast of Australia, but the form that grows in Tasmania is actually critically endangered and is very distinct genetically to the mainland varieties of this species. We were growing this as a seed orchard in our nursery and realised that our neighbours at Government House had a mainland variety of this growing in their gardens, and they very kindly removed it for us so that we could actually keep the integrity of our seed orchard and our seed collection, which was very kind of them. Some other considerations: how much space can you commit and for how long? We're finding that the majority of our seed orchards now will be in the nursery for between 6 and 8 years, some more than that. So it takes a considerable amount of time to build up not only the genotypes but the plants themselves to generate those good seed harvests. Other resources that need to be thought about is how you actually harvest the seed. Who's going to be doing it? A lot of seed will want to be shedding and needing to be harvested during the summer periods when there's a lot of holiday periods for people. So relying on volunteers might be more difficult than imagined. You also need to be able to adjust and adapt accordingly. You will find that some plants, you just haven't got the right conditions to grow them within your nursery, and you need to think outside the square in how to achieve a good seed harvest or a seed orchard or an ex situ collection whereby they can be maintained outside your nursery facilities. And some years ago we were involved in this project on Macquarie Island. There's a species of cushion plant there called Azarella macquariensis. It's critically endangered due to a dieback that's going through the wild populations on the island. We knew very early on that we would just not be able to grow this species at the Botanic Gardens facilities. We hadn't got the right conditions for it, and we knew that because we had tried to grow them in previous years. So it was pointless bringing back plants to try and grow here at the gardens. So we decided to set up our seed orchard and ex situ collection remotely, actually on the island itself, but away from the infected populations in the wild. And so we've got a seed orchard and seed and ex situ conservation collection there, and that's been running since 2010, and it's still going now. So need to think outside the square. Other thing that you might need too, that you might not think about, is you might need something other than horticultural skills. Here is our previous nursery supervisor, Michelle Lang, rapidly sewing up some netting to generate some seed harvesting bags for us. And here we are. Another danger that's inherent within seed orchards is that you can go a little bit doolally in bagging or harvesting seed. So I mentioned that understanding the biology of your species is really important. An example of that is this little species, Tetratheca gunnii, or Shy Susan. It's interesting in that it has a very small flower that needs to be buzz pollinated by a native bee. Now you guys on the mainland are very lucky in that you have native bees that form colonies and hives, and you can actually manipulate them and move them around places. Here in Tasmania, all our native bees are solitary, so we're not able to have a hive and bring one into our nursery area to be able to pollinate species for us. We have tried to hand pollinate Tetratheca gunnii over the years, and it's incredibly time consuming. So other ways that we've tried to work around this is that we have taken the plants to gardens that have high native bee populations when things are flowering, leaving them there for a few days and bringing them back. That's been relatively successful, but we have also been working on trying to augment our bee habitat within the nursery facility itself and encourage the native bees to come in and stay in the nursery and do the pollination for us. Ways that we've been doing this is by creating habitat under our benches, as you can see in some of the images here. We've also tried to create nest material and nest areas habitat wise for the native bees to come in and stay within the nursery facility. Another interesting aspect of the biology of Tetratheca gunnii is that it has an elaeosome on its seed, which means that it's very attractive to ants, and it's actually ant dispersed. So that was something else we needed to think about in terms of managing the risk of losing our seed to ants during the harvesting period. I have to say that I think the most useful tool to have when managing a seed orchard is the ability to observe what's happening with your plants. And a classic example of this one is this particular species, Verbalium devisii. We'd been growing this species since the 1990s, just as an ex situ conservation collection within the nursery, but decided to do a seed orchard on it. We had assumed that as the plant flowers and sets seed over many, many months, that what we would do, we would sit the plants in some trays, and the seed would drop into the trays, and we'd be able to get our harvest that way. The first year that we did this, we ended up with only 100 seeds in the trays, which was terribly disappointing. The following year, we decided to observe the plants a little bit more closely, and we found that the seed capsules actually had a really explosive dispersal mechanism, and so the seeds were being shot out of these capsules, which is why they weren't dropping into the trays the previous year. So by bagging the entire plants for a period after they'd finished flowering, we managed to harvest 35,000 seeds after that. So it's a really good tip. Observe your plants. In closing, I don't think I need to tell anybody watching here today that time is running out. Climate change is having a real impact on our biological diversity across the globe right now. The latest IPCC report highlights that the window of opportunity to do something about it is closing rapidly. However, the good news is that with plants, we have the ability to capture their biological diversity in seed form and store it for future generations. We just need the opportunity, the resources, and work together to get the job done. Thank you for listening today.
Thanks so much, Lorraine. And what a fascinating presentation. There are so many things to consider if you're setting up a seed orchard, aren't there? Remember that you can ask questions in the Q&A section at any time, and we'll put those questions to the presenters shortly. Next, we have a fascinating insight into the collections of the Australian National Botanic Gardens from Dr. Zoe Knapp. Zoe is the Conservation Manager at the ANBG, overseeing nursery horticulture and data management to support their living plant collections and collaborative conservation projects. Zoe's team applies specialised horticultural skills to investigate the intricate propagation and growing requirements of native plants. Her team works with other scientists and conservation agencies to understand aspects of plant biology, including population genetic structure and the requirements for reintroduction to the wild. Zoe's background is in plant ecology, academia, environmental consulting and public policy. She's worked in the Department of Agriculture, Water and the Environment for over 10 years, supporting effective partnerships, science, natural resource management and strategic policy. Welcome. Thanks so much, Zoe.
Thanks everyone. Thank you for the opportunity to share some of our work today. It's really exciting. Just share my screen. Where is it? Sorry. Great, so I'm really excited today to share some of the work that we do in the ANBG nursery and living collections and how that contributes to conservation in Australia. I thought today I'd just start by sharing a little bit of context about the Australian National Botanic Gardens for those who might not be familiar. So we're located in Canberra, not so sunny Canberra today, on Ngunnawal country, and we're on the slopes of Black Mountain, which is actually right on the edge of the city. And we're right next door to the CSIRO and just across the road from the Australian National University. So it's a pretty convenient location. It's quite unique for a botanic garden in that it's actually managed as one of our 6 Commonwealth terrestrial reserves, which is overseen by the Director of National Parks. So that includes some of our iconic parks such as Uluru-Kata-Tjuta National Park, Kakadu, Norfolk Island National Park, Christmas Island and Budary National Park as well. So we have an interesting connection to national parks and islands across the country and some pretty fabulous opportunities for field trips, I won't lie. And we're managed underneath the broader portfolio of the Department of Agriculture, Water and Environment. So a federally managed botanic garden, which I suppose is a bit unusual. Within the Botanic Garden itself, we've actually also got the National Seed Bank and the Australian Biological Resources Study, which is a unit that compiles and shares taxonomic information about our Australian native organisms. And whilst they're not technically associated with the ANBG also hosts the Australian Seed Bank Partnership and the Australian Network for Plant Conservation, which we're really lucky to have that close proximity and many partnerships with the ANPC. We also, as I mentioned, are right next to CSIRO, and we have a very close association with the CSIRO through a joint venture called the Centre for Australian National Biodiversity Research, and that also includes the Australian National Herbarium. We're located in the ACT, and so we also have very close relationships with the ACT government, and then being in southeastern New South Wales, have close relationships with our New South Wales Department of Planning and Environment, and also our regional botanic gardens and conservation NGOs. And we really value our partnerships, and we'll highlight some of those today. And the Botanic Gardens, or the ANBG, is also somewhat unique in that its focus is entirely on native flora. And we also hold 24% of threatened species that are listed under our federal legislation, the Environment Protection and Biodiversity Conservation Act. I just wanted to share too a bit of context about what we're discussing today, which is the increasingly important role of botanic gardens in conservation. And a big part of the reason why we have such an important role is because we effectively have a collection of living laboratories that are either on site or within close partnerships that allow us to provide end-to-end solutions for conservation. So right from understanding a species, describing a species, understanding its identification and its evolutionary relationships, being able to collect seed and understand germination cues and bank the seeds, then understanding the propagation techniques, which is not always an easy thing. Because of the focus of native flora in the ANBG, we have a lot of horticultural expertise on how to grow various native flora, but also for many threatened species, and that includes some quite difficult species to propagate and cultivate as well. And what I'll highlight shortly is we can also get plants back into the wild through our conservation partnerships. And also, as highlighted earlier today, we can establish many different types of insurance populations, which includes plants growing in our living collections and our seed production areas. And then we also have a permanent nursery collection. And before I move on, one of the things I just want to highlight too is that one of the real values of our collections is, as John highlighted earlier, is that they are underpinned by scientific data. And so all of our collections will be attached to information about where they've come from, how they've been grown, what types of research we've done on them. And for most collections in the Australian National Botanic Gardens, they're actually directly linked to a voucher herbarium, a verified herbarium specimen in the Australian National Herbarium. So if any changes happen in their taxonomy, we know about it pretty quickly. I think what's most astounding to me is that I think there was a bit of discussion earlier from John about how conservation was often in the remit of the scientists, but actually, astoundingly, the nursery and living collections have a huge role in this end-to-end conservation cycle. We contribute to everything from field collection. We provide plant material from the Australian National Botanic Gardens for other research projects outside the gardens or with our Centre for National Biodiversity Research. We learn how to propagate and cultivate these species. We have our seed production. I've got a picture there in the top right of our seed production area with some plants growing in there. We have many conservation partnerships. We're really involved in education and awareness, which is a really important aspect of what we do. And as I mentioned, we have some permanent nursery collections where we hold insurance populations as well. And the picture in the bottom right is of a small snapshot of our permanent nursery collection. So I just wanted to share today some of the work that we're doing in collaborative conservation projects. I've said here that we have 18 projects underway, but this number changes all the time, and that depends on how you define a project and the scale, and plants are coming in and out every day, which is really exciting. And a lot of what we do is to investigate the propagation and growing requirements of native species, which is not always very easy, and that can be by various methods, different hormonal treatments, lots of other factors that can influence that success. We grow a lot of plants for translocation efforts, and we also hold many insurance populations in our ex situ collections, and we produce seeds in our seed production area. And on top of our collaborative conservation projects, we're also holding many other species for future research and conservation efforts as well, or just as backup collections. So I just wanted to share a couple of our case studies as examples of the different types of contributions that we're able to make to conservation. So the first one is Muhlenbeckia tuggeranong, or the tuggeranong lignum. This is the ACT's rarest plant. There's thought to be only a handful of these plants remaining now along the Murrumbidgee River corridor in the southern ACT. And interestingly, near a popular swimming spot that I've probably swum past many different times. Also interestingly, this plant was actually first discovered by Dave Mellinson, who was a horticulturalist at the ANBG, and he found the plant while he was on a weekend fishing trip along the river. And the photo on the right-hand side, the far right, is a plant growing in a native population in the wild on a rock, so it likes this sort of rocky slopes and riverbank slopes, and that photo was taken by Murray Fagg about a week after the species was first discovered in 1997. The species is also interesting and challenging for conservation because it's dioecious. So Lorraine just highlighted the challenges of this particular characteristic. So with separate male and female plants, and with so few plants remaining in the wild, this can really limit breeding options. So it's thought now that there's actually only male plants left in the wild, and these plants have not been observed to produce fruits or seeds in the wild at all. So you might be wondering then, looking at my pictures, well, what's that picture second from the left? Isn't that a picture of fruits? Well, that's what I'm very excited to share. So in 2015, our nursery manager, Joe, actually visited the site with the ACT government. So Emma Cook has been leading a lot of wonderful work on this project, and just had the initiative to take some cuttings at the time and brought them back to the nursery. Well, it turns out that has resulted in us holding the only female plants remaining of this species in our nursery. And so we hold lineages that are now extinct in the wild, and that was also shown through genetic research undertaken by Linda Broadhurst at the Centre for Australian National Biodiversity Research. So we're so grateful to have that partnership to be able to do the genetic research to better understand our species and conservation. And that has actually guided how we manage these ex situ collections. So we're now maintaining each of the known lineages of the plants. But just in the last few months, we have some really exciting news, and that was that Pete Riddell, our senior nursery horticulturalist, had noticed that there were some flies hovering around the plants, and subsequent to that, the plants had started producing fruits. And so Pete collected a bunch of the fruits off there and took the seeds out, and some of those have gone now to the National Seed Bank, and we're working now on some germination trials both in the nursery and in the seed bank. But we are excited, exclusive as to this meeting, that we have just achieved the very first seedling of this species ever seen since the species was discovered. It's very hard to see. There's a tiny, tiny little green leaves coming up in the pot there, but we're just absolutely overjoyed about this success. So our goals now for this species will be to continue to maintain each of the known lineages in insurance collections. We'd like to establish some research to better understand the breeding strategy and ecological requirements of the species, and also hopefully to get it into our seed orchard and start producing some more seeds and grow some new seedlings. We have another big successful project on at the moment. This is a huge partnership project called Survive and Thrive. It was established through the National Parks Conservation Trust, which is located at the Botanic Gardens with us as well, but is actually an independent organisation. And many other partners, which include the Worldwide Fund for Nature Australia, World Wildlife Fund Australia, Australian Alpine National Parks, ACT Government and New South Wales Department of Planning and Environment as well. So we have a lot of partners involved, and our goal is to protect bushfire affected species in subalpine regions across Namadgi, Kosciuszko and Alpine National Parks. So everybody will remember the 2019-20 bushfires for quite some time to come. And it was thought that many species across the region had been burnt across their entire range. And so we've chosen 5 target species and a broader list of 12 to investigate what's remaining. So do some new population surveys across the different alpine national parks, and then to investigate seed biology, propagation techniques, to try and create some new seed collections, and to really develop all of the techniques that are needed to be able to establish populations for recovery efforts in the future if they are needed. So here's our team at the bottom. There's Tamara on the left in the field, Pete second from the right, doing some cuttings, Beck third from the right, who's leading this project, and she is moving some plants from the polytunnels there as the cuttings strike roots. And on the right is Viola impressera, the dwarf violet, which is one of our 5 target species. So far we've had some really early successes. This project has just started this last summer. We've done some new field surveys which have revealed 2 new populations of the viola species that we didn't know about before. So that's really exciting news. And the team has been busy. We've managed to get all 5 of our target species into our nursery and propagation with some various trials, which I'd be happy to talk about anytime. And interestingly enough, we're expecting these all to be quite difficult, and some have been. The viola here is quite easy to reproduce. The team has discovered that you can just literally pull the plants apart, so it reproduces by division, and put them in new pots and it grows like crazy. A little bit unexpected. It also grows quite easily from leaf cuttings as well. So we still have another field season to go, which we hope to collect a lot of new material and seed, and we're working very closely with our national seed bank, and we're so lucky to have that very close working relationship with them to continue understanding the requirements of these species for recovery efforts in the future. I love highlighting this species, Swansona recta, the small purple pea. It's so difficult to grow, actually. It's become a bit of a dirty word in the nursery. Oh no, not more Swansonas. But we are excited to keep working on these collaborative projects with the ACT government and also with New South Wales Local Land Services. So the Swansona recta is a small perennial herbaceous species that was once widespread across southeastern Australia, from northern Victoria to central to northern New South Wales. But it now occurs in just a few smaller fragmented populations. There's a southern region around that ACT, and then a northern population area around the New South Wales Central Tablelands. And so our goal is to try and augment some of the existing populations to boost the vigour and the genetic diversity of those populations. And one of the ways that we've been doing that is to establish a seed production area with some plants growing in there. So there's Beck in the far left in our seed production area. She's putting some beautiful seed bags. There's a Tom North from the curator of the National Seed Bank's design these seed bags. I always say I would have gone with hot pink, but I also love this colour. And she's placing some bags over the seed pods, and then we'll wait for those to dry and collect the seeds and take them to the seed bank for processing. And so when we established this seed orchard, it was established in a scientific design to maximise genetic diversity. So from all of the populations across the ACT region, they've been intermixed in the seed orchard to make sure that that maximises outcrossing among the different populations to try and boost diversity. And that's based on genetic research that was undertaken also through the centre. And what we've noticed, some really interesting findings so far, in that the germination of wild collected seeds is quite low. It's only around 10%, but germination from the seeds we've collected from our seed orchard at the ANBG is markedly increased, so we can get 50 to 60% germination rates, which is a really exciting finding. We're growing a lot of these plants in the nursery at the moment. This is a few of them, the third picture from the left. And then whilst they do produce very beautiful flowers, we have separate projects on the southern populations and the northern populations. And because of some genetic distinction between those two regions, we are maintaining those the genetic integrity of those different collections by cutting the flowers off. So it feels a little bit like Morticia Adams deadheading the roses, but it must be done in the name of science. So this has been a really successful project so far, and the team has done a huge amount of work to figure out how to grow quite a difficult species. And we look forward to getting more of these out in the wild in the coming years. Another project that we have going on is Eucalyptus recurva, the Mongalo mallee, which is probably Australia's rarest eucalypt species, with only 6 plants remaining in the wild. It's historically had poor seed set in the wild and a low propagation success as well. So the species is a dense mallee shrub endemic to just a small area of New South Wales, not too far from the ACT. Interestingly too, this was first discovered when a landholder brought a specimen to the AMBG, excuse me, in 1998. It was first thought to be an Eriostemon species, but The AMBG's botanical ecologist at the time, Mike Crisp, took a closer look and discovered it was actually a eucalypt. Excuse me. So it's been historically difficult to propagate, but in March 2020, the AMBG and the New South Wales Department of Planning and Environment collected a few seedlings and have managed to have some initial success. So the 3 plants growing on the right were grown from seeds just a couple of years ago. So it's great to see them getting so big, and they will also provide material now for grafting trials. So here's Pete on the left doing some grafting with his grafting tools. And there's a plant in the middle that's just being grafted. We're quite fortunate through our partnership with CSIRO to have had some seeds donated of a Eucalyptus recurva hybrid that they've been studying. So we've been able to use that as the rootstock. And it appears that we're having some early success in grafting this species. There's so many factors that can influence the success of these grafting trials. And so we're just really lucky to have the right horticultural expertise to be able to undertake these projects. And our goal ultimately is to establish grafted plants from each of the 6 wild plants in our seed production area, and hopefully to produce lots of seeds, increase genetic diversity, and get the species back into the wild. We have a lot of other projects going on. So with our connection with national parks, we've got a project on the Christmas Island fern, and there's a really nice case study that the team has put together in chapter 13 of the Germplasm Guidelines. So please read that if you're downloading the guidelines. There's also an article that's out now in the latest edition of Australasian Plant Conservation, written by Catherine Scobie, who's a senior horticulturalist in our team, about some of the work that we do as well. We also grow lots of plants for research. Just in the middle here, there's some plants here that might be new to science, and we're just waiting for those to be formally described by our partners at the Australian National Herbarium. We do a lot of work for translocations, but also for habitat restoration for animal species as well. We're involved in bushfire recovery, the Tropical Mountaintop Species Project that was mentioned earlier. Excuse me. And as an aside, we try to maintain our own AMBG living collection as well. Thank you.
Thanks so much, Zoe, and thanks for persisting. I know you're recovering from being unwell, so I really, we all feel for you. And look, there are so many people in the chat and online who are really excited about your success with the Muhlenbeckia, with the Tuggeranong lignum. So, so well done, well done. We now have a short video from the ANBG team to address one of the underrepresented groups in conservation collections, that is the ferns. Capture of fern germplasm and spore banking was of great interest in our FENA conference on the environment last year, and we're so pleased to have one of the chapter authors guide us through the process today. Fern spores can be stored in a similar way to orthodox seeds, though their short lifespan means that drying, processing and storage must occur quickly to maximise viability. Tom North, the seed bank curator at the Australian National Botanic Gardens, will take us through the collection, handling and drying process for fern spores. And their techniques are described in detail in chapter 13 of the Germplasm Guidelines. Thanks so much, Tom.
Non-seed-bearing plants can include bryophytes and pteridophytes like mosses, hornworts, liverworts and fern. These types of plants have spores instead of seed, and conserving them can be straightforward or require specialist techniques. In this video, Tom North from the Australian National Botanic Gardens, Canberra, shares their techniques for conserving fern spores.
So in terms of conservation of non-seed-bearing plants, it's really important that we understand at what stage those plants are at in order to be able to collect the material that we're interested in conserving. So mostly it's easier if we're collecting spores, and for that we need the plants to be at the sporophyte stage where they're producing sporangia and the spores are encapsulated within the sporangia. I've got a fern frond here with frondlets and pinnules on it that's got sporangia on the back of it. So what we're looking for is just to make sure that those sporangia are mature and ready to release the spore, and that they've actually haven't already released, or that they're too green and immature. So I'm just going to have a look. So we've got sporangia on the margins of these pinnules here that are just about ripe and ready to collect. So I'm just going to sterilise secateurs. The other thing we need to do is make a record of what we're collecting, where we're collecting it, who collected it and the date, and give it an individual collecting number. Now to collect into, I'm using just a heavy brown paper bag. We wouldn't use plastic because of the risk of building up static and trying to get spore out of that, but also the risk of cultivating mould on the spores or the fronds that we've collected. We're just going to collect the frondlets. So we're only taking about 10% from this, and then a sample across all the ferns in the population.
Fronds are kept flat between 2 clean sheets of paper and secured in a plant press for drying over 24 hours. Sporangia on the frond will dry within 24 hours to release the spores if collected when ripe.
So the next step is then to just sieve that collection through quite fine sieves. What I've chosen here is 2 sieve sizes, just to get rid of the extra large, any frond material that's still there, and then a smaller sized sieve under that to just collect any of the sporangia material that's going to go through as well. So it's just a light sieve and tap through there, and that spore should all go down through that top sieve, and I've got just a little bit of material there left on the top that's non-spore, and again that can be discarded. And then with the finer sieve, again it's just tapping down and then checking as well under your Maggie lamp or a microscope, just to check that what is left in that sieve is the sporangia material or debris and not actually spore. And your spore should all be in your lower or collection tray at the base there. The spore that I've just processed there looks like it's non-chloriferous, which means it's got quite orthodox storage characteristics. We'll dry that at between 10-25% relative humidity. So you've got a couple of options there in terms of storage. Probably the preferred method is to go into a glassine envelope where the seed can dry down, the spore can dry down to appropriate level. But at the moment, I'll transfer this into a glass vial to go into our dry room for a couple of days to desiccate to appropriate levels. So just to do that, and just tap that into, so and we'll leave the lid off that in the dry room so that it has a chance to dry down.
Drying time varies depending on the type of spores you are collecting. For non-chloriferous spores, it can take between 5 and 7 days. Chloriferous spores can take between 1 and 3 days. You can find further information about drying times on page 407 of the guidelines.
Once it's been in the dry room for a couple of days, we'll assess moisture within the collection, but also test the viability. before we then shift that into minus 20 degrees.
All the information about collection and conservation of spores is included in Chapter 13 of the Guidelines, which can be downloaded for free.
Fantastic. Thanks so much for that, Tom. Now I'd like to invite Lorraine and Zoe to join me for questions. We have time for just a few questions before we take a break, before getting into the rest of our presentations. Thanks so much, Lorraine. Thanks, Zoe. So, Lorraine, a question for you first. You mentioned that nursery space is quite a big factor in accommodating the seed orchards that you're setting up. Is there a way that you're working to address this issue in future?
Yes, we've got quite a small nursery here in Tasmania, and it's hard to juggle how many seed orchards we can cope with. So interestingly, just a couple of years ago, we were approached by the Australian Plant Society, the Northern Group, who wanted to try and help us with our seed orcharding program. And so we set up a trial project with them to grow a viola species and see how they went. Up in their area, they had a secure nursery that was possum proof, and so it ticked a lot of boxes for us. The Australian Plant Society, many people would be members of that society themselves or know of that group it's been a very long-term group across Australia. The skills within those organisations are amazing. So they took on this seed orchard for us, and I'm really pleased to say that just last summer, they did a seed orchard collection. They had 14 volunteers over the Christmas period, and they managed to get us our largest collection of violas that we have ever achieved. So, you know, I think it was about 92,000 seeds. So it's pretty good. Very successful. So we're hoping that will continue into the future. They're very keen to do more for us.
Yeah, fantastic. That sounds so good. And the pollinator baiting sounds really interesting. John Arnott was wondering, is there any potential to send plants to mainland gardens to establish orchards? I know it's always tricky to move plants around in that way. And Zoe, maybe there's places outside of the ANBG's facilities where, you know, an effective seed orchard can be set up too. Lorraine, did you want to answer that one first?
Yes, I'm sure partnerships is going to be a big thing into the future. Definitely, we'd probably look at that. But we all know the Botanic Gardens are all struggling to do their own state species. So we'll see how we go with that. But yeah, definitely open to suggestions there.
Yeah. Okay, fantastic. And Zoe, I'll see if I can turn your camera on for you. Let me see if I can do that. No, it's not working at the moment, but we can hear from Zoe. Sure. That's okay. Chris, maybe you can try and turn Zoe's video on for us if you're available. We'll see what we can do. So Zoe, I'm really interested in how you refine the methods for propagation at ANBG. And also, so do you have an experimental setup for the species, and how do you capture and share that information relating to propagation?
That's a great question, actually, Amelia. Thank you for asking that. So I suppose what is amazing, I think, in the team is that there really is a scientific approach to everything, and there's not always enough material to do it in a really robust data capturing way. But the team does take nonetheless a very scientific approach in what they do. And usually what they'll do is start with looking at what the taxon is and what sort of conditions that taxon would normally require to be propagated or cultivated, and start from there, and then trial a few different things that might work. And then for capturing the information, so we have a really comprehensive database that we call IBIS, and it connects with the Australian National Herbarium and the Living Collections database as well. So we capture all of our information there about our species provenance, accession numbers, how we're growing it, what different research we do in the seed bank, and the herbarium voucher specimens as well. And as part of that, we're able to capture information about propagation treatments that have been tried by the team. over time, and then we can pull that data out later. But something that we're also working on more now is to try and publish more of those methods as well. So a big goal for us is to share that information more widely. And Beck Highland, one of our horticulturalists who's leading the Survive and Thrive project, is, as we speak, building some fact sheets that we can start sharing more of this information, which is really important.
Yeah, fantastic. That's excellent. That's excellent.
Going.
That's okay. That's all right. And so one quick question so we can give everyone a short break before we kick off again. Jens is interested in whether anyone is trying to grow the Euchre curva using tissue culture. So have you had a look at that yet, Zoe?
So our team were really fortunate to visit Mount Annan Botanic Gardens last week. I feel like it's been a long week. And so we're really grateful to Mike Elgie, who's on the line today, for being a wonderful host and showing us around. And that actually, just being there, gave us an opportunity to talk about some different opportunities for propagating this species. So we're definitely looking, and so thank you, Jen, for making that comment, because it's really valuable to share those different tips and techniques. And the team is definitely looking into some other options, but that's just the value of these partnerships that we have and the opportunities to talk through forums like today.
Yeah, fantastic. Thanks so much. Well, thanks so much to all the presenters this morning and for being available for questions. All of the presenters that you've seen will be back at the panel session at the end of the webinar today. So we've now got a short break, so feel free to get up and have a stretch or make a cuppa, get some lunch while you enjoy a short slideshow. We'll look forward to seeing you back online at 12:30 p.m. Eastern Standard Time, 10:30 Western Time, or 2:30 p.m. in New Zealand. So we'll have a couple more presentations before our panel discussion. See you all soon. Welcome back to our Plant Treasures webinar on the role of the nursery and living collections in plant conservation. This morning, we heard about current practices in nurseries and living collections and some of the challenges associated with maintaining both the diversity and quantity of Australian plant species in cultivation. This afternoon, we look in more detail at future needs and we get involved in a panel discussion with today's presenters. The possibility of pest and disease incursion is always a challenge in maintaining living collections, and we're lucky to have our next speaker, Amanda Shade, who can talk about managing living collections both in the nursery and in the ground. Amanda is the Collections and Development Manager for the Botanic Gardens and Parks Authority based at Kings Park Botanic Garden. In this role, she oversees the production of mostly Western Australian plants for use in the Western Australian Botanic Garden, Kings Park amenity parkland areas, bushland restoration and threatened species translocations, as well as overseeing the curation of living collections and germplasm held at the Western Australian Seed Centre at Kings Park. Welcome, Amanda. Just take yourself off mute, Amanda.
Yep, sorry.
Oh, good.
Yep, got it. Thanks. All right, let me just share my screen. No, it's this one. Okay. Okay, can everybody see that? Is that okay, Amelia, or are my notes just being displayed as well?
That's lovely. Well done, Amanda.
Brilliant, excellent. All right, let's get started. Thank you everybody for listening to this. So as Amelia mentioned, I am going to be talking about biosecurity considerations in living collections and coming to you this morning or afternoon for most of you from Whadjuk Noongar country in southwest WA. So first up, I guess, what is biosecurity? So broadly, it's the management of the risk of animal and plant pests and diseases either entering, emerging or spreading within a location in order to protect the environment, the community and the economy. So this presentation is going to cover elements of biosecurity in relation to managing and curating living plant collections. And while the focus is primarily going to be on nursery collections and in-ground botanic garden collections, the main principles that I'm hoping to cover can be applicable to other kinds of living collections. Biosecurity is quite a big subject. I realise probably too much to cover comprehensively in this short space of time. So to make sure I don't go off on any tangents and cover the important points, I'm going to break it down into a couple of different elements. Firstly, talking about implications to existing collections within nurseries and botanic gardens, and then also managing incursions of new biosecurity risks to those collections. And I'm going to use some examples or some case studies from recent experiences that we've had here in Western Australia. So first up, talking about implications with current collections. I've broken it down into 4 little subcategories there, probably more than familiar to most of you, but I'll just briefly touch on them. So quarantine, very important to have a level of control over what new material is coming into existing collections and where it's being sourced from. If there are any uncertainties or concerns about the sources, quarantine is essential. It's really imperative not to introduce any new pathogens or weeds via plant material or soils into existing collections. A period of monitored quarantine is usually appropriate to determine if there are going to be any issues, but a quarantine setup is also useful for any material that needs to be separated from a collection due to suspected infections or for closer monitoring, for example. Quarantine areas ideally should be located away from the main collections and production areas where access can be restricted. And access is another important consideration when dealing with biosecurity. This is much easier to do with a secure nursery facility, a little bit trickier when dealing with botanic garden in-ground collections, particularly if, like us, your botanic garden is not fenced and is open 24/7. There are some challenges associated with that. The second dot point there that I wanted to talk about quickly was hygiene. Everyone is probably more than familiar with the importance of hygiene. It is essential for all horticultural activities. Strict hygiene protocols are one of the main preventative actions to safeguard against introduction or spread of any biosecurity threats. And specific accreditation programs such as that run by the nursery industry or Green Life Australia, I believe they're now called, with their nursery industry accreditation scheme. They can provide specific guidance on hygiene practices that are really important for protecting valuable living collections. Ongoing monitoring and assessment is also really important. Collections can be kept for multiple different purposes, depending on different agendas and research programs and what have you. And often species within collections are being grown and maintained well outside their comfort zones or out of their normal environmental range, in modified environmental conditions, or restricted to container growing. Some things just don't enjoy that at all. And these things can affect the growth and the overall health. So consistent monitoring and assessment of performance is vital. And when I say it can affect the growth and overall health, that's not necessarily always in a negative way. Sometimes some things grow exceptionally well outside of their comfort zone and could in fact themselves become a weed potential. So we really need to consider that too. And the last thing I wanted to talk about was record keeping, which really goes hand in hand with all of the above. And it's really important for planning, for interpreting any test results, for developing and trialling seasonal preventative treatment programs, and also understanding which species are potentially vulnerable to the pathogen that you perhaps are dealing with. Biosecurity considerations for living collections can also be supported by strategic documents such as collections policies and collections management plans. I want to use a little example of an unfortunate incident that we experienced here at the Kings Park Nursery. So this slide is of some plants affected by powdery mildew. Everyone's probably familiar with that, certainly people who work in nurseries, but we really noticed this becoming a significant problem, not just in our nursery collections, but also some of our botanic garden collections, perhaps about 5 to 6 years ago. And this corresponded with a significant increase in humid conditions in Perth. At the risk of sounding like a typical West Australian, we have a dry heat over here. We're not used to humidity in Perth, but particularly during those change of seasons at the start of autumn and spring, and we hadn't experienced that before. So the first year that it became a noticeable problem, it really took us by surprise. And despite all of our monitoring, our record keeping, all of those things I just spoke about on the previous slide, we actually weren't ready for it. And it wasn't one of our regular pathogens. It wasn't on our radar. We hadn't experienced it on the scale that we were seeing it at. So, and because of that, we actually suffered quite damaging losses in susceptible species, and that included some conservation collection species as well, unfortunately. Admitting that, I know it seems like a bit of a rookie mistake, but we have used it as a really valuable learning experience. We now undertake a seasonal regime of preventative spraying to combat any further losses, and that's been really successful. We've also used this opportunity to consult with biosecurity specialists in this pathogen. We now have a much better understanding of how it behaves, the species that are most susceptible. And we've also been working with a plant pathologist in our local biosecurity agency here in WA to gather important data, for not just for his research, but also to work together on different ways of managing future incursions. And from a nursery perspective, it's no great issue to move anticipated susceptible species into quarantine areas, restrict their access for their management. But in the botanic gardens or other in-ground collections, that's a slightly different management process. And we've been just monitoring susceptible species. Obviously, we can't rip them out of the ground and move them to areas to monitor and treat. But for some accessions that are particularly affected, we have to rethink completely how, or even if we continue to include them in our planted collections. Another quick case study, and this hopefully will be a good example of how biosecurity is approached differently in wider landscapes as compared to nursery collections. Most of you are probably familiar with this pathogen. This is malaria. It's another biosecurity concern we've been dealing with here for a number of years. And it's significantly altered how we manage our existing garden collections, but also how we approach developing collections in certain areas of the park. Like a number of pathogens, it's all but impossible to completely eradicate. So we need to look at an effective management, monitoring, hygiene, and other practices to minimise its impact. So what we've done is we've comprehensively mapped detections of armillaria throughout our botanic garden and parkland collections. So theoretically, we can predict where we will see it fruiting every year. We're learning a lot about how it behaves and what we can modify in the greater environment to manage its impact. We know we think we know the ideal conditions for the fruiting bodies to appear, and we're always ready and on the lookout as that time approaches, and it's getting pretty close to that time of year. All of our horticultural field staff are fully trained in how to recognise it, how to report the sightings, how to deal with the fruiting bodies, and also appropriate hygiene protocols so we can prevent any further spread. We also have certain machinery and equipment access restrictions, and they're put in place seasonally to avoid any cross-contamination of infected and non-infected areas on the gardens. So that's all well and good, and that has contributed to significant improvements, but there are additional challenges that are faced when we are dealing with high profile or historic collections, such as this one pictured. This is an established memorial tree avenue in the main drag of Kings Park. You can see the city of Perth in the background there. It's a major thoroughfare. It's the main entrance into the park. It's very popular and well-trafficked area, and these are memorial trees, as I mentioned, and you can see in the bottom right there, that is a scar that has been caused by armillaria that we picked up a couple of years ago. Quite alarming. So the way that we have managed this infection, so we've looked at more preventative measures. We've tried not to be too reactionary. So traditionally, these avenues were surrounded by turf. They were heavily irrigated. There was a lot of mowing and aerating and coring and a whole lot of other associated turf activities, very highly trafficked areas. So late last year, we undertook the removal of a significant amount of the turf surrounding the infected tree and its neighbours. We introduced a layer of organic mulch. We reduced the irrigation. We gave applications of things like ferrous sulfate and sea salt and acidifying fertilisers. Unfortunately, we also have some pH issues with our groundwater. And we will do this at appropriate intervals as well. So armillaria season is fast approaching here in Perth. We're carefully monitoring this area to see what impact these changes in the management practices have had, because this is a very significant collection of trees, and we really do not want to lose them. So management of existing collections from a preventative perspective is quite achievable, but there's a lot of unknowns, and sometimes we think we might know what things we might get incursions of, and sometimes they completely take us by surprise. So this is where surveillance programs or structured surveillance programs are really important. And I wanted to just talk about a couple of programs that have had widespread botanic garden participation in recent years. And the first one is one that has been run by the Botanic Gardens Biosecurity Network, and that is managed through Plant Health Australia. And there's another one that is run by the International Plant Sentinel Network through Botanic Gardens Conservation International. So the first one, the Botanic Gardens Biosecurity Network, so that's been running over the last 2 to 3 years, and participating gardens have undertaken lots of training, and they're targeting 5 pests of concern. So gardens that were involved in this have chosen sentinel plants within their gardens to, and that's based on the known susceptibility to the particular pest and consideration of risk pathways such as high traffic locations and other environmental factors. And what we did is each spring and summer, we would do monthly monitoring and surveillance and submit reports on a monthly basis via a My Pest Guide reporter mobile app. And this is one of our horticultural field staff inspecting a camelorsium for any signs of myrtle rust. The IPSN monitoring program was more interested in surveillance and reporting on selected Northern Hemisphere species that are being grown in Southern Hemisphere gardens. And that was for them to learn about what kind of problems may be encountered in these different environmental conditions. So gathering data on biosecurity issues in different environments is really useful in developing a greater understanding of how plants may respond to different pathogens in different conditions. And this slide is of an English oak that we are growing. We have a collection of English oaks here in Kings Park, and as you can see, this one has got quite a bit of mildew. So following on from that and going back to the Botanic Gardens Biosecurity Network program, so one of the 5 pests that we were monitoring for was Polyphagus shot hole borer. And I'd like to just, as a bit of a case study, share a recent experience with this particular pest because it is new to Australia. And I just want to highlight how this incursion has impacted both the way we currently manage some collections here, but also how we will now need to modify and develop the management plans for future consideration. So very briefly, and just to recap, I suppose this pest, the shot hole borer, was first confirmed in Perth about 9 months ago. It was reported by a private garden owner. And they reported it through the My Pest Guide Reporter mobile app to our local biosecurity agency. It was on an exotic tree, although it has a very wide host range that includes both native and exotic species. It was fairly quickly identified as Polyphagus shot hole borer, and the biosecurity agency here sprang into action following their protocols for responding to a suspected new incursion. Subsequent surveillance revealed further detections on other nearby properties. They did the whole quarantine area implementation, and information was circulated widely through industry and the general public. And we were informed of this fairly early on in the process, and we began conducting our own surveillance throughout our collections within the Botanic Garden and our nursery as well, with a focus on the known host list as had been developed by other parts of the world where this pest has spread, Israel, California, South Africa. So this was one of the 5 pests, as I mentioned, that we had been reporting on for 2 years via the Botanic Gardens Biosecurity Network program. And our sentinel trees for this pest had been carefully chosen. They were thoroughly checked every month. We had no evidence of detection at all. We had not found it. So after comprehensive surveillance within Kings Park by our local biosecurity agency, it was unfortunately detected here within the park. However, the specimens that they found it on were not thought to be serious contenders for hosting this pest, and they were in quite inaccessible parts of the park. It wasn't a location we would have considered as a risk pathway. And some species that are being recorded as badly infested in other parts of the world, we're not really seeing evidence of that here yet, although we do realise we are in the early stages of the incursion, so that may change. We're also noticing that despite being described as lazy, this borer can certainly move quickly, and I'm just going to see if this video will work. So it actually can move quickly when it wants to. And also for some distance. So, you know, the information that we received was that it would only travel about, 30 to 40, maybe 50 metres, but we are seeing that that is not the case. So previous information was also telling us we would be likely to see them in branch unions of trees. We're not seeing this. We are seeing the majority being found in deadwood or tissue that's been previously damaged by other means. So in other words, we're seeing some really different behaviours under Perth conditions than reported elsewhere. So this experience highlights just how important good data is in understanding how pathogens behave in different situations. And while it is still very early days with this pathogen, as we develop more information and build data on this pest, we can make more informed decisions about how to develop management plans for some of our collections that we already have or that are impacted or are likely to be impacted. This is very much a work in progress. As I said, it's early days. There's still many unknowns. It has reinforced, however, our strategy to develop adaptable rather than reactionary responses to surprise incursions, because this was a surprise. Oh, whoops. Sorry, I didn't mean to do that again. So I noted earlier that collections can have numerous different purposes, and wanted to touch briefly on one very important role of collections, which is, and others have already mentioned this, but it's about providing important ex situ protection for threatened species or those from threatened ecological communities. So biosecurity threats to natural environments can result in more species requiring safeguarding within collections, which may mean extending existing collections or developing new ones or duplicating at alternative locations, which John actually touched on earlier as well. This is often more effective if there is preparation time to safeguard the species that are experiencing threats to natural populations. And as an example of that, myrtle rust, which has been touched on by a couple of speakers already. So that's yet to be detected here in WA. And because of that, that's really given us some crucial lead time to undertake preparedness field collections to boost current holdings of vulnerable species. And we're about to head off north to do that very thing. To finish up, I wanted to share some really useful resources for anyone who might be interested in looking further into this subject. I've probably already referenced most of these throughout this presentation, but just to recap. So Green Life Australia's Nursery Industry Accreditation Scheme, again, really good for hygiene guidance and support in nursery situations. We have Plant Health Australia, and their Botanic Gardens Biosecurity Network and the International Plant Sentinel Network. So these organisations and programs have very informative websites, often social media platforms that are really good as well. They offer access to technical information, webinars, training opportunities, and other really useful links. Plant Health Australia have also updated their National Plant Biosecurity Strategy 2021 to 31, so that's worth a look as well. And also in each state, there's local biosecurity agencies that have a range of information and support available. Our local biosecurity agency here has been really amazing in working with us and collaborating on our shot hole borer incursion in particular. And as mentioned already, networks such as BGANZ and AMPC. And that's it. Thank you for listening.
Thanks so much, Amanda. And I'm sure there's lots of people who are watching today who would appreciate that need for lead time, some time to prepare before we have to take action. Yeah. Next up, we have a really interesting presentation from Dr. Sally Norton, who will talk about what we can learn from conservation of crop wild relatives. Sally is an Australian plant scientist and national leader of the Australian Grains Gene Bank, with over 20 years experience in the collection, conservation and curation of agricultural plants, specialising in crop wild relatives. She's travelling today, so she's kindly recorded a presentation for us. Thanks, Sal.
Good morning, everyone. Thank you for the opportunity of presenting for Plant Treasures today. I unfortunately can't be with you today, but Kath Whitehouse has kindly offered to sit on the discussion panel later if there's any questions. What I intend to talk about today is to give a bit of an introduction on the Australian Grains Gene Bank and really around what we can learn from the conservation of crop wild relatives. But before I launch into that, I'd like to acknowledge the traditional owners of the lands in which we all meet today and pay respects to their elders, past, present and future, and extend that respect to Torres Strait Islander and Aboriginal people present today. So really the Australian Grains Gene Bank is an agricultural gene bank. We have the mandate to acquire, conserve, maintain, distribute grain crop genetic resources, so germplasm, for the benefit of the Australian grains industry. This is important for Australia because 95% of our agricultural plants actually originate from overseas, and the majority of what we grow today has been adapted and bred to meet Australia's conditions. The AGG, established in 2012, at the time it was the newest gene bank, agricultural gene bank in the Southern Hemisphere, and we opened in 2014 in Horsham. We operate in the public domain, which means we operate under the International Plant Treaty for plant genetic resources for food and agriculture, and we operate within the global network of agricultural and conservation seed banks. As I said before, we're for the benefit of the Australian grains industry, so we definitely have a national focus, although we have quite a lot of international engagement and are reliant on overseas gene banks for access to material. So a bit more about us. We have tropical and temperate crop species, and that covers the cultivated varieties that are grown in farmers' field in Australia today and from around the world. We have landrace, which are village varieties, which are very diverse. And we have quite a large proportion of breeding lines which have been developed for assessment of suitability to Australian conditions. We also have crop wild relatives. We cover the entire agroecological zones of Australia, from cold temperate right through to monsoonal tropical, and everything in between. We are a seed bank, so we're conserving orthodox seeds that can be stored at the -20 degrees temperature we have here. And at the moment, we have around 187,000 accessions, and we're growing exponentially at the moment with the deposits of a lot of material coming out of programs in Australia. You can see on the right here the panel which shows that probably just over half of the collection is winter cereals, and the rest of it's made up of temperate and tropical pulses, oil seeds, legumes and other crops as well. We're very diverse. We've got over 2,000 different species, including subspecies, and we have origins of over 150 countries. So our core functions in the gene bank are no different than any other gene bank around the world. We acquire material, so that's through post-entry quarantine or crop wild relative collections. You can see a couple of pictures there of some recent trips that we did to the Northern Territory. Our post-entry quarantine facility, we have 2 programs within Australia. We have one at Horsham and we have one at Tamworth in New South Wales, which is really important to have that compliance to federal regulations for importing from overseas. Our storage and duplication, we operate at -20. We also back up into the Svalbard Global Seed Vault all of our unique material. Maintenance of our collection is what I'll talk a little bit about today. It's viability testing and regeneration. This is probably on top of the acquisition of material and the importation. The maintenance is the bulk of the work that we do in terms of labour and resources. We do viability testing for around 6,500 accessions per annum. We're regenerating currently around 4,500 different types of material each year, but that will increase over the next 12 months to closer to 5,000 with some new infrastructure coming online. The distribution is our client function. We are distributing around about 30,000 different samples a year all over the country, about 350 unique clients or recipients, and some of those are within the same organisation, but a very broad coverage of Australia. Our volume of distribution has doubled over the last 5 years, so there's increasing demand for a lot of material, particularly around crop wild relatives of Australian and overseas origin, and also diversity sets that are being developed for specific purposes. Around 90% of our distribution is done internally to Australia, so to organisations within Australia, and about around 10% is exported, but that export is actually increasing at the moment. Gene bank management, large part of that is around data management, documentation, and those relationships, partnerships and collaboration that are essential for us to achieve what we are planning to achieve. And one of the fun things that we do is our outreach. We have an education program called Get Into Grains. We regularly host artists within the gene bank itself, and we are being involved more and more in a lot of capacity building partnerships with Australian Seed Bank Partnership, Millennium Seed Bank Partnership, Crawford Fund, Crop Trust and others. And that's for people to travel to Australia to learn from us and us learn from them, and also for opportunities for us to travel overseas as well. So the maintenance of the gene bank collections, what I mentioned earlier, is a very large part of the resources and labour that we have within the program here. I just wanted to break down a little bit about the complexity we have across the Grange Gene Bank. We have such a broad diversity of species, ranging from cultivated right through to wild relatives and tertiary gene pool wild relatives. So they're a little bit more challenging. So for cultivated species, they're generally or relatively straightforward. Often one regeneration environment, such as what you're seeing on the screen there, is suitable for a wide range of species. So the top panel there, you can see some pulses being regenerated in a shade house. You've got some lentils, you've got some peas, field peas, and you've also got some chickpeas. There's also cereal on the other side, outside of that screen. The maintenance regime is fairly similar. And down the bottom right there, you can see one of our field increase trials of wheat and barley there. So we can do a big mixture of things there. They're generally very easy to germinate. They've got relatively simple viability testing protocols. And for nearly all of these cultivated species, including landrace species, they can generally be sown direct into the soil. So it's very, very straightforward and quite simple. There's complexity around that though, but generally quite straightforward. The in-crop care, so the pest disease regime, nutritional regime is similar across many species, so it gives you the opportunity to plant multiple species in one location. And generally, there's very little shattering of seed, which is important because it means we don't have to bag and you can just harvest the material as it goes. When we look more towards our crop wild relatives, they're incredibly challenging. So we have specialised protocols for species, and sometimes that can drop down to subspecies level, which makes it even more challenging. Specific regeneration requirements, which are linked to specific environmental requirements. Often we have to mimic very closely the environment from which these species originated in. And that can be temperature, light regimes, soil, nutrition, biota, the list goes on, and humidity as well. The germination is often very difficult and very often it's complicated. And for the collections that we make that are fairly new, there's often unknown protocols on how we might actually germinate these ones successfully, and often there's limited seed. So we're taking an educated guess on related species. Most of our wild species cannot be direct sown for regeneration. They just do not germinate. So often we're doing pre-germination before sowing, and this is particularly evident for secondary and tertiary gene pool species. The in-crop care, they vary across species, and a seed often shatters. So we can co-locate species together, but there's very different management protocols which need to be done. So we've got to be very careful on how we do that. If we look at Australian native sorghum species as a bit of an example, and this is a case study out of the Germplasm Guidelines. The natural environment for these native sorghum species is generally subtropical, annuals and perennials. They germinate after the wet season and set seed between March and May generally. Although there is one temperate species in the New South Wales and Victorian Highlands, it's perennial and it seeds around August and November, and that one is quite challenging to actually get to set seed, although we're having more luck here down in Horsham than we used to in Queensland. They need the extremes of temperature, so they have quite cool nights and quite hot days a lot of the time, certainly for the northern ones. The southern ones probably more cool. They're often growing in nutrient poor soils. They all shatter their seed and they all outcross, so we need to control their pollination. So for Australian native species, if we look at the germination protocols, cultivated species, very straightforward, 25 degrees, constant temperature between wet filter papers or rolled towels, and most of the viable seed is germinated within 4 to 5 days. And you can see this panel here under the cultivated here, which has a bit of a diversity of seed size, shape, colour there, but all generally very simple to work with. Australian native species, on the other hand, require a striated temperature of 25 and 35 degrees and light regime for 16 and 8 hours each and a 14-hour day length. We excise these seeds from their seed coverings. You can see on the top right panel there, the Australian crop wild relative seed types, large awns, very tightly held. Glooms are very tight, holding the seed within the structure there. Very difficult to germinate, a lot of dormancy in there. And to break that, we have gibberellic acid at 1 millimolar. And unlike the cultivated species where you know very quickly whether they're viable or not, usually 10 to 12 days, most of the viable seed will have germinated. The panel on the bottom right is from the Germplasm Guidelines, 3rd edition. You can see here the bottom couple of lines here are native species, Australian native species, Macrospermum and Brachypodum, grown at 25 constant temperature, and you can see that they're barely hitting 5% germination there, barely. Whereas if you do the striated temperature regime and excise them from the seed coverings, they jump right up there into that sort of 70 to 90 up here, which is in line with cultivated species if you do them at 25 degrees or in the striated temperatures. So you can see that the difference in germination from just changing the protocol, which is not unexpected. So the regeneration of these species is also quite challenging. As I mentioned before, pre-germination and transplant, you can lose quite a few plants in the transplant process. They need very loose draining soil low water and nutrition requirements. Pollination control, top panel here, you can see that the crispy bags we have on here. That's to control their pollination, but also to capture the shattering seed. These things are terrible shatterers. The bottom right panel shows a mixture of cultivated species and landraces here, so all within sorghum bicolour, direct soil sown, little dormancy, pollination control, because they are outcrossing, but it's a paper bag on top that you harvest all in one go, and they're generally non-shattering. My experience with the native species when I first started working with them, treating them like the more wilder type bicolours, they just were very poor. Pulled them out of the glass house, pulled the drippers out, put them outside for a bit to figure out what to do with the next batch, and then they thrived when they actually started drying out. So very, very sensitive to conditions. So the main things I think I wanted to cover off on today was what are the lessons we've learnt from crop wild relatives. They are incredibly challenging in every aspect, from the collection in their natural environment. So you can see here a couple of images. So the top middle one here, that's of wetlands coming into Kakadu National Park. All of the rice is the light coloured grass, which is mid-panel there. And there's an awful lot of lily pads, water, and potentially crocodiles between where we're standing and where the rice is. So a bit of a challenge. Sometimes we manage to get it on the side of the floodplains, as we're seeing Gibson do at the bottom there. Then we've got some wild pigeon pea as well. Pods at the top left, down the bottom you can see the slope that we're up on trying to find that. The conservation of these species, although they're orthodox, they are generally shorter lived with lower viabilities. Initial viabilities, germination often complicated, so it takes more time. The regeneration takes a lot more time and care and attention to detail to make sure that we're producing good viable seed. Highly labour intensive compared to our cultivated species. And often when we're making new collections, there are many unknowns of the phenology, physiology, and they're highly variable. Different species within sorghum have quite different requirements. And one of the things that we're learning from trends and also from the knowledge, more of the knowledge of the diversity of these species, that they're going to be critical for the future of food security. And for me personally, they're the most rewarding to work with. The cultivated types are actually quite boring compared to these, so it makes it a little bit more interesting as we're moving forward. So in closing, I want to thank you to the AMPC for the opportunity to present. Thanks to my team, partners and collaborators who all work so incredibly hard to conserve and maintain our germplasm, which is going to be essential for future food security. And just a note that the AGG is a partnership between Victorian Government and the Grains Research Development Corporation. Without their support, we would not be able to do what we need to do. So in closing, thank you again, and I will hand over to the next presenter, and then thank you and enjoy the panel discussion. See ya, bye.
Fantastic. Thanks so much, Sal. Sally's colleague from the Australian Grains Gene Bank, Dr. Kath Whitehouse, is going to join our panel session this afternoon. So please ask any questions you have about crop wild relatives there. I think there's really a lot we can learn from their approach in regenerating so many seed collections in their nursery each year. We now have our final video for the day, which was filmed in the beautiful Kings Park and Botanic Garden in Western Australia. It begins with researcher Dr. Emma Dalziel talking about all the different ways that situ collections are used. Then Dr. Leonie Monks and Dr. Andrew Crawford from the Western Australian Seed Centre take us through the translocation process for the threatened species Grevillea caliantha. We also get an insight, as we have throughout the day today, into why collaborations and partnerships are critical for conserving Australia's national plant treasures. Enjoy the video.
Plant germplasm is any living collection of plant material that represents the genetic line of an individual or species. Globally, collections are increasing in number and diversity. It's important to understand then why ex situ collections are made and how they will be used in the future.
So there are multiple uses for ex situ germplasm. First and foremost is obviously conservation. We can also use it for ecosystem restoration, plant breeding programs, or for horticultural programs. In places like Kings Park, where are Botanic Gardens, the germplasm can also be used to grow plants for display, which is an important component of public outreach and education. Depending upon why we collect germplasm in the first place will dictate how we collect it in the field and then what kind of material we collect as well. So for example, when we're collecting for conservation programs, we often need to collect maternal lines from individual plants. But then for other end uses, such as plant breeding, we might be interested in particular forms. So plants with different flowering colour or flowering time are of interest.
One of the most frequent conservation end uses is translocation. That is returning plants to the wild, either as new populations or bolstering existing ones. In this video, we are going to learn how an ex situ collection can be used for a conservation translocation.
Translocations are about establishing viable populations of plants in the wild and ones that have the genetic diversity that allows them to adapt and persist when conditions at the translocations change.
The Western Australian Seed Centre have been conserving Grevillea caliantha with the long-term goal of restoring wild populations.
Well, Grevillea caliantha was listed as threatened way back in the mid-90s, so it's known from 6 small populations up from the Dandaragan area, which is about 150-200 kilometres north of Perth. Most of the populations were only known on road verges, and it was challenging to manage these populations in the long term. So translocations then became one of the most useful recovery actions we could do to make sure that this species had a chance of actually persisting in the long term in the wild.
Before commencement, the team needed to develop a translocation plan, which detailed the how and the why of the project. They also needed to review permits, approvals, hygiene risks, and exit strategies. Then they could start wild seed collections based on their decided sampling strategy.
So for a conservation seed collection, we want to make sure that we sample widely both within a population, but we also want to make sure that when we're sampling a species that we sample across the populations. And we want to make sure that that sampling is random. But for other purposes, maybe for plant breeding, you might want to make considered decisions when you're selecting for forms or flower types or those sorts of things. If you have populations that are quite disjunct, you then need to make a decision about whether you can mix those populations in your translocation. In the case of Grevillea caliantha, all of the populations occur in a relatively small area. It was decided that all of those populations could be mixed when a translocation is undertaken.
Once in storage, propagation methods can be tested and include vegetative or seed germination.
For rare species, often we don't have a lot of seed to be able to try lots of treatments in order to work out the optimum way of germinating something. So we might use surrogate species to work out our methods and then apply it to our threatened species so that we're not using up that limited seed source.
Trying to establish how best to germinate or propagate Grevillea caliantha was done through trial and error and also using background information that people already knew about how to germinate or how to propagate Grevilleas in general and other species that also occurred in the same family as well.
Once the living collection is established, reintroductions or translocations can occur, but that doesn't mean the extra collection is no longer required. Often, an experimental or staged approach is needed.
We've been trying different methods of establishment as we've gone. We don't go too large at any one step of the process, so that if for some reason things don't go right, we still have more seed and opportunities to come back and repeat that process in the future.
So after we had planted them, we installed a watering system to make sure that the plants could be watered over the first 1 or 2 summers. And then also we installed fences to ensure that they didn't get grazed by herbivores such as kangaroos and rabbits. The other important thing that we do when we put plants out there is we tag them. and we ensure that we record all the information that we already know about those individual plants, and then we can follow each individual plant at the translocation site. So we have actually had quite a reasonable number of plants survive, and they've produced flowers and seed. We've had very limited recruitment. We know that recruitment is probably not likely to happen in large numbers in the absence of fire. Some sites are doing better than others. So as we learn which sites are better for establishing grevilleas, we're focusing more and more on those sites. And then we're thinking about, well, what are our next steps as well? So success comes in many forms. It's not just understanding whether the plants survive, but it's also other aspects of their ecology and their life cycle.
Conservation collections are a long-term commitment and need replenishing. Often, fresh collections will be sourced from the wild, but it isn't always an option.
In the case of Grevillea caliantha, some of our seed collections represent populations of the species that are now extinct in the wild. So in that instance, regeneration is really the only option. The next step in that translocation process will actually be to establish a seed orchard to bulk up seed of that species. But regeneration can pose problems for a long-lived perennial species that may take a long time to reach maturity or may require a very large area to grow an adequate number of plants. So in those instances, we would then try cuttings. So some plants can be successfully grown from cuttings. We can still get a good genetically representative sample to use for those translocations.
Translocation is one potential end use. The decisions at project commencement about intended use will inform everything that follow, including collection, propagation and storage methods. But there are always unexpected challenges and uses, which is why partnerships are so important.
Partnerships and collaborations are absolutely essential for starting and maintaining ex situ collections. Conservation often doesn't receive a lot of funding, and we require support from public and private corporations to support the work that we do. Organisations such as the Australian Seed Bank Partnership, which really put together a coordinated Australia-wide response to ex situ conservation, and ensuring then we also have the duplication of material across Australia. And then finally, for research using ex situ collections is often multidisciplinary and requires a great deal of expertise across a wide range of different areas. So including plant biologists, soil biologists, or even people who work with animals.
Detailed information about utilisation of ex situ germplasm is included in Chapter 15 of the Guidelines. The FloraBank Guidelines deal specifically with seed sourcing and ecological restoration. The Translocation Guidelines can be used to plan and undertake best practice translocation.
Thanks so much to this afternoon's presenters, Amanda, Sal, and the team from the Western Australian Seed Centre. Please keep your questions coming in as we get ready for our panel discussion. We're going to have a quick Menti poll now to get our panel discussion started and to hear from you. So bear with me while I share this poll. Okay, should be able to see that now. All right. So this morning I shared a graphic from the guidelines that linked ex situ collections to in situ management of native plants. We know the botanic garden sector has really specialist skills and experiences that are important to maintain germplasm collections. They are also skills that can be used to support natural populations, either currently or into the future. So now we're going to get your feedback on this graphic using a Menti poll. Remember, you can respond using the Menti app or the web browser and entering the code on the screen. So the code for this poll is 85544439, or you can use the QR code on the screen. We can see people joining now. Give a few more minutes, a few moments for people to join up to the poll. And then the code and the instructions, except for the QR code, then stay on your screen for a moment as we address these questions. Fantastic. And remember, the videos that we've shown today and the recording for this webinar will be available later on the ANPC YouTube channel. So don't forget to check back and have a look at our resources there. All right, we'll get stuck into the poll. So for this question, we'd like your answers in 1 or 2 words so we can create one of those nice word clouds. What skills do you use? What skills do you see that your colleagues have to support conservation, both ex situ and in situ? They might be skills in handling plants or in handling the data associated with collections. Maybe there's some particular skills and experience you noticed during today's presentations that are used to really shape those ex situ collections and to support plants in their natural habitats. Some botanic gardens also have herbarium collections, so there's taxonomic and plant identification capacity. And you can submit more than one answer here if you wish. Fantastic. So some great responses coming up there. Propagation is huge, isn't it? All of those skills around multiplying and growing our plants, figuring out the germination requirements, knowledge, passion. Passion is huge. That's one of the fabulous things about talking to people about plants and going into the nursery and visiting botanic gardens. Seed collection, data collection, looking after the data is really important. And we have a chapter on that in the guidelines as well. In the nursery, gentle hands. Oh, very nice. Very nice. Being adaptive, yeah, looking at what needs to happen and how we're going to make it happen. Yes, weeding, always weed removal. This is fantastic. What a really nice collection of skills there. And then all the associated skills with those other ex situ collection types, tissue culture and things like that too. Fantastic. Okay, the other thing I'd like to find out, and this is the last poll for the day, are there any actions that we've missed in this graphic? Actions that botanic gardens can take to support ex situ collections or in situ populations? These are free text responses, so you can submit a short sentence here or more than one answer if you'd like, and I can make that graphic a bit bigger so you can see. If we were going to redo this graphic, what else would you like to see in here? And we expect to see a really wide range of possibilities, given all the different activities that happen in botanic gardens. Public education is huge. That could go across all of those activities, couldn't it? Yeah. Seed orchards, quarantine, yeah. More communication between gardens and maybe even sharing of species, fantastic. Skilled and professional, yeah, definitely. People with all those skills. Funding, yeah, long-term funding is always a challenge given the resources that we know it takes to start and maintain these collections. Online resources, maybe a database for public viewing, definitely. Training, nursery, collection duplication. Yes, we've come back to that quite a few times today. Duplicating seed collections at different seed banks and also duplicating living collections in that meta collections kind of strategy. Student internships, really nice one. Yeah, bringing people into the industry, training them so we can all share our knowledge. Indigenous cultural knowledge, yeah, it's an area we absolutely have to do better. There's so many different ways of understanding the landscape and looking after country that we need to bring all different kinds of knowledge together. Absolutely. Fantastic, such good responses, and you can keep those responses coming in as we kick off into the panel discussion today. Thanks so much for your responses there. Fantastic. So we'll use these results as we evaluate the germplasm guidelines and plan new activities within both ANPC and BGANZ to support plant conservation in the future. I'd now like to introduce our facilitator for the panel session, BGANZ Council Representative for New South Wales, Michael Elgie. Michael commenced his horticultural career nearly 20 years ago as an apprentice, and during his career, he's worked for the Ryde TAFE, the Ryde School of Horticulture, the Office of the Secretary to the Governor-General, the National Herbarium of New South Wales, Royal Botanic Gardens Sydney, Rockhampton Botanic Gardens and Kershaw Native Gardens. Since September last year, Michael's been the Curator Manager of the Australian Botanic Garden Mount Annan. He's an active member of the BGANZ community and currently represents the BGANZ New South Wales Regional Group. And Michael's also represented the Queensland BGANZ Regional Group between 2019 to 2021. Welcome. Thanks so much, Michael.
Thank you very much, Amelia. Good afternoon, all. And first up, I'd just like to acknowledge the traditional owners, and I'd like to, of all our nations. And I'd like to acknowledge that I live, I work, and I'm zooming in from the traditional lands of the Darrell people. So how great is this webinar so far? I just want to say thank you to AMPC, BGANZ and the Ian Potter Foundation for their support to make sure that this webinar happened today, and Amelia, of course. So today we have 6 contributors to this panel with a range of backgrounds and expertise. On my screen, I'll go from what's on my screen left to right. I have Catherine Whitehouse, I have Amanda Shade, Andrew Crawford, Lorraine Perrins, John Arnott up there. Now, for a bit of background, you can probably see there's 2 new faces that we haven't spoken today, so I'll give a background on Andrew and Catherine. So Andrew Crawford is the manager of the Western Australian Seed Centre in Kensington, Perth, which is part of the Department of Biodiversity, Conservation and Attractions. Andrew has been involved with collections of Australian native plant species for over 30 years, with 20 years spent collecting seeds from Western Australia's conservation significant plant species. Andrew's work covers all aspects of the seed conservation, from seed collecting, processing, storage, testing, and generating plants for translocation projects, as well as seed-focused research aiding in threatened species management and recovery. Catherine Whitehouse is a research scientist at the Australian Grains Gene Bank. Catherine is one of the few international experts in applied seed longevity research with a vast real-world experience in long-term seed storage for crop genetic resources conservation, where effective utilisation of the germplasm in the principal goal. Catherine's research focuses on optimising species longevity under long-term dry storage and improving the efficiencies within gene bank operations. So today we've heard about the current practices in living collections, such as establishing seed orchards and bringing threatened species into collections and managing pests and diseases. We've also considered future needs, including preventing diseases, incursions, the need to regenerate collections coming out of seed banks, and all the resources involved in those processes. We've also heard about the specialist skills that we have to support conservation of native plants, both in situ and ex situ. So we'll move to the panel discussion now. So if there is anyone that has any questions, can they please put them into the chat, and I'll try and manage the panel discussion from this rowdy mob and read the questions and somehow tie it all together. Since we actually haven't heard from Catherine or Andrew, I'm going to start with those 2. So the first question for today's panel discussion is for Catherine. So Catherine, if you just want to unmute your microphone there. How can living collections increase the genetic diversity?
Can I answer it in the form of seed?
Absolutely.
Okay. So at the Gene Bank, we conserve all types of seed, and they all vary in different levels of genetic diversity, and they hold many different traits. So over time, plants have been modified through cultivation to suit our needs. And with that, we've lost a lot of the original diversity. So the wild relatives, which we also conserve at the Gene Bank, they're the most genetically diverse, and they've evolved to survive tough conditions, and so are considered already better adapted to pressures such as those from climate change. But also, as they're genetically related to the cultivated types, their genes can be transferred to increase the genetic diversity that's been lost from the cultivated gene pool. So it's important for us at the gene bank for us to assess the genetic potential that we have in our collection. And we do that by characterization and image-based phenotyping, as well as genotyping and sequencing. So we feel if the gene banks are used correctly, they can actually accelerate the role of evolution.
Wonderful. Now, the second question, I do see a few questions starting to pop up in the chat, which is good. But for the second question for today is for Andrew. Welcome, Andrew, and sorry, welcome, Catherine, as well. I forgot to say welcome to you both. Andrew, is there a need for flexibility within our nurseries, within botanic gardens and like institutions to have capacity to be flexible when a crisis occurs? And if so, a bit of elaboration on what you think that flexibility needs to be.
Yeah, most definitely. And I guess to me, there's two main areas that would cover. So I manage a conservation seed bank, and one of the main things that we're storing is seed of threatened species. And I guess the ultimate aim of those collections is to be able to translocate those species back out into the wild. And when we do those translocations, we would like to aim for a population size of about 250 plants. So that means we need a lot of seed often to be able to get those 250 plants. And unfortunately, for some of our most threatened species, getting those quantities of seeds is a real challenge. There's one critical species we've got here over in WA, Divisia. We've been collecting seed of it periodically for over 25 years, and we've got less than 1,000 seeds. So, it shows the challenge. So one of the areas, for that flexibility is to be able to, as Lorraine was sort of pointing out in her talk about the need for seed orchard, seed production areas, to be able to easily or readily boost the quantities of seed of some of these species, to be able to undertake meaningful activities back out in the wild. And then the other area I see, the need for flexibility is, unfortunately, often the funding and resourcing for seed-based work is out of sync with the on-ground work of translocation and the like. So if we're collecting seed of a particular species and we want to do our routine testing in that, we're generating germinated seeds, which means plants. And ideally, we want to be able to use those for to use them. But often there's, as I say, there's a sort of a separation between the funding. We need resourcing to be able to do those plantings. So to have that flexibility of capacity, which obviously includes resourcing and space and all of those issues, we could better utilise what's sort of a byproduct from the seed banking process.
Thanks for that, Andrew. Yeah, space, especially with the nursery aspect, is always a big struggle for a lot of mature botanic gardens. And I'll probably throw this question over to Amanda, talking about living collections, following on from Andrew's question. Our living collections, they serve a purpose to display plants for education and conservation projects. From a biosecurity point of view, what strategies or processes would you recommend to have in place to help protect those plants from be it internal or external pressures of bringing potential pathogens into a site, or our green spaces at the moment are really valued and should be really valued by the community and getting used a lot more. So we're having more chances of incursions of biosecurity risk into our living collections in botanic gardens and arboreta. What strategies do you have in place at Perth over there, Amanda?
Well, there's a number of things. I think public education can't be understated. And by what methods that happens, it's up to each state, I suppose. But making people aware that, they can't just wander off into the bushland with muddy boots, or they can't drive their cars in different areas, just things like that. And not just saying you can't do it, but explaining why. the implications. And a lot of the biosecurity risks, it's not just the natural environment that there are ongoing issues with. It's, you know, there's industry implications, there's commercial implications, all of those sorts of things. So they actually have a human cost as well sometimes. So I think public education and how that is directed is really important. From a more localized perspective, you know, restricting access and monitoring, surveillance, you know, treatment should be the very last thing, really. If you've got all of the hygiene protocols in place, the less you'll have to do, I suppose. You know, quarantine, as I mentioned in my discussion, is vital. You can't just bring anything in from wherever. And having that kind of documentation as a government department, you know, we've very well documented. So having, you know, things in place for contractors. You know, we have a lot of events here in our botanic gardens, as I'm sure a lot of you do as well. And we have very strict guidelines for people who want to bring equipment in, who want to bring material in, plant material, all of that sort of thing. So having those things in place, but not being so regimented that you just say, no, you can't do that. Actually explaining to people the repercussions of what will happen if they don't follow those rules, essentially.
Yeah, education, even from those polls that Amelia just hosted on the survey, education is the key to, I think, a lot of our conservation messages, from our living collections that are within the botanic gardens to our bushland or conservation sites, to our seed sites, to biosecurity that impacts everyone, probably far more than what a lot of the population actually understand at the moment. And there is a question in the chat which is regarding communication. So I'll open it up to everyone on the panel to answer if they wish. And it's Denise from the Macadamia Trust. And I'll read out what Denise has said. Cross-pollination with cultivated species is a major challenge for conserving 4 macadamia species. The Macadamia Trust are addressing this by growing material from cuttings, but this is quite a difficult message to get across in the community of conservation, to the community or the conservation community, when seeds are so easily accessible and readily able to be germinated. What advice from members of the panel here do you have for the Macadamia Trust? John, can I pass that one to you?
I was just going to suggest that Lorraine's example with the Canidia was kind of a good one, to have that direct discussion with people proximate, and that worked okay for you, Lorraine, in terms of avoiding that sort of genetic pollution?
Yes, it did. As soon as we explained what we were doing, the other side of the fence, they were more than happy to remove their plant for us for that period. So yeah, communication's really key. But with the project that Denise mentions, I think it could be a little bit more complex because it's probably over a larger area. So we've spoken about education more broadly, but maybe just getting out there a bit more about what your project is about in local newsletters or local gardening groups, all that sort of thing. Even the friends of your botanic gardens can be really good at disseminating that information out.
And maybe it's a little bit like what we were talking about earlier, Lorraine, with, you know, moving from the site to another site where those pollinators are present, and or the hybridizing pressures aren't there. So maybe it's, you know, go to the Botanic Garden Network and look to do some of that propagation outside of natural range, outside of cultivated range of plants that are potentially going to pollute that genetic resource. Just a thought.
Thank you, John and Lorraine. Yeah, genetic pollution, for lesser word, is something that can be considered for all of us that manage living collections and part of the conservation projects. Lorraine, I know I haven't picked on you just yet, so I'll forward you a question from that we've got here. Where do you see botanic gardens positioned into the future for undertaking seed orchards, be that small within nurseries or large-scale seed orchards as part of conservation projects? And do you think it's a viable option to put all that pressure onto Botanic Gardens, or do we need to evolve somewhere in that space?
Well, there's such a lot of work to do. I don't think Botanic Gardens can do it all. We've mentioned numerous times funding is a big issue, resourcing space, it all becomes quite difficult. So by developing the networks that we're trying to do throughout the country, I can see that, from the speakers today, there's lots of networks out there. We need to engage as many people as we can to try and get on board with assisting us. But I guess when you're talking about lots of people doing lots of projects, it's the management of those projects in keeping in with the standards of the Germplasm Guidelines and how to do it properly that is difficult. And that's where I think Botanic Gardens come in to assist people with that and help get that advice out there and get that standard across the board. Because if we're going to do it, we want to do it properly and make sure that we get good quality collections stored in our banks. I don't know if anybody else has got anything to add to that.
I'll pass it over to the panel. Does anyone else have any input to that question regarding seed orcharding into the future and the role of Botanic Gardens? Amanda? No? Okay.
I will just add, actually, just one other thing I will mention is that we have done remote seed orchards, like I mentioned the one about Macquarie Island, and that was an option for us. Some people have mentioned the fact that some plants do better outside their range, some plants don't do so well within it. So we're actually looking at remote seed orchards within Tasmania as well. So some of the alpine species we can't grow in Hobart. So we're looking at perhaps having video monitored seed orchards elsewhere that we can monitor from afar and use organisations that are close to them so that we're sort of overseeing it, but they're monitoring it for us as well as an option.
Yeah, that's awesome. Over to you, John.
Lorraine, did the satellite seed orchard up with the AMPC in the north of Tassie, was that a success? Did that work?
With the Australian Plant Society, yes, it was incredible.
Oh, sorry, APS, sorry.
Yeah, it was successful. And we only just a few weeks ago had a review meeting with them up there and spoke to all the volunteers, and they thought it went really well. And they were very, very keen to do more. And we started off with a very quick seed orchard for them. Violas can get done in about 18 months or so. So we thought we'd get a good quick one for them to trial. But they're actually interested in doing some of the more longer term ones over the 5 to 6 years as well, which is quite encouraging for us as well.
Thank you for that one. I might pass this one over to Andrew. Is there a danger of over-reliance on translocations as a solution to pressures such as land clearing?
Okay, well, I think here the important thing to remember is I don't think translocation should ever be seen as a solution to the threats. We shouldn't be using them for mitigation. So it shouldn't be seen that, you translocate a species, therefore it would be okay to clear the natural population. The primary focus, I think, should always be on preserving the species in the wild. And that's going to be the simplest and hopefully the best approach. We always use the translocation as a supplementary tool. Often, the natural populations, the threats aren't manageable, so we need some sort of a backup out there. The other thing to remember with translocations is we've been doing them for a long period of time, and to actually show that a translocation has been truly successful may take decades or that sort of thing. In the video you watched that the Grevillea caliantha, we now have more plants in the translocations than in all the wild populations put together. But those populations, we can't say they've truly been successful because we haven't had those subsequent generations. they're establishing a good soil seed bank and that, but are they going to be able to be self-sustaining? So translocation isn't a quick fix. It's a long-term proposition, and yeah, shouldn't be seen as a offset measure. It's a complementary strategy.
Thank you for that, Andrew. I think everyone here agrees 100% with your response to that question there. It is all about complementary. Catherine, I've got a question for you in the question answers. This is from Karen Somerville. Have you seen much variation in germination difficulty or longevity among different provenances of crop wild relatives in your work?
Yeah, well, the wild relatives, they're always really hard to germinate. And a lot of it's guesswork on how we get them to germinate, because most of the protocols we don't know. I haven't actually looked into it personally, but the germination is surely variable depending on where they naturally occur, I would assume. We can learn a lot from gathering information on the environment that the seed develops in and the timing of when they usually release the seed. So seeds which usually develop and shed during colder weather, they may need, for example, frost or chilling treatments before they will germinate. Longevity also varies. There's been correlation between seed that usually originates from hot, dry climates are generally more longer lived than those that originate from cooler, wet climates. In general, wild relatives are pretty short-lived. Don't know why. I haven't worked a lot on wild relatives. Most of my work's on cultivated, but I have a theory that it might be because wild relatives generally have a longer developmental time compared to cultivated species. So like the timing that they flower and set seed is usually sporadic and can sometimes be spread over quite a long period. So this makes it hard for us to pinpoint when seeds are at their maximum like harvest maturity or physiological quality. And if you harvest outside of that time, that it really affects how long they can live in storage. So that would be my theory on why they're generally shorter lived. It's probably because we don't really know how to access them at their peak optimum time.
Yeah. Working and trying to manage living collections and seeds and biological organisms, we think we know a lot, and then we get thrown curveballs and we think they're going to do A, but instead they do X, Y, and Z instead. I'm pretty sure that will go across for a lot of people here in the work they're doing, the conservation of plants and other species around the world, or across Australia, sorry. Question for John, and I might make this the last question because we've already gone over time. And this is around, adapting to our living collections, and we don't always know what they're going to do and how they're going to act in the future. So how has a living collection strategy in your gardens, John, changed over time?
Yeah, thanks, Michael. It's really interesting at the Cranbourne Gardens. That the Cranbourne Gardens was started in 2006, completed in 2012, and it was really set up as a horticultural display garden. So conservation, horticulture, ecology, research, they were aspects of the garden, but not primary focus. And when we did a review of our master plan in 2016, it said that we should actually start to address that and start to build some of these layers into the living collection, almost reverse engineer that botanic or conservation horticultural agenda into an existing display garden. And that's happened over the last 5 or 6 years in reality. But it was one of those examples where the strategy was there to say, let's start to build conservation horticulture, ecological collections, have that agenda reflected in the Australian garden. So there was a strategy that was sitting there, but it was actually driven by staff, and it was driven by the passion of staff to actually identify this as an opportunity and really have a crack at delivering it. So there's been a transformation in recent years at the Cranbourne Gardens from a major focus on horticultural display to a really serious introduction of conservation, horticulture, ecological collections into the collection, driven by strategy, fueled by people. But the other interesting thing which has happened at the Cranbourne Gardens is that integration of science into horticultural operations and utilising horticulturists in support of the science agenda of the RPGV. And to that end, we've got a fellow called Russell Lark, who's team leader of horticulture. Russell Lark through secondments and mostly a secondment, and there was a studentship. He was embedded into the science team up at the Melbourne Gardens, and actually started to form some really meaningful relationships with the science team. So, yeah, it does come down to relationships, collaborations, individuals, and I think the Cranbourne Gardens is just an example of the commitment of a fantastic team of horticulturists to recognise the opportunity to have a crack at it.
Thank you for that, John. That's really good to hear, because at the moment here at the Australian Botanic Gardens Mount Adam, we're going through the whole process of redoing our living collection strategies at this moment. But like you said, and I think it's relevant for what everyone has participated and listened today. You know, we're all here because we are driven by passion for change, and we do want to conserve our plant species, not just here in Australia, but we want to see it happen worldwide. And there is a few other questions in the chat, which I'm not going to get to, so I apologise to everyone there. I will try and cut and paste some of these and get them to Amelia and see if we can get them answered from the panel remotely in due course. So I'll hand it back over to Amelia, and thank you. And thank you to the panel. Greatly appreciate your time.
Thanks so much to all the presenters and panelists today. Well done, Mike. Great discussion session. And thanks to you, our audience, for your participation today and for contributing to the discussion about the importance of living collections and conservation horticulture. If you're keen to explore this topic further, I encourage you to think about attending the 7th Global Botanic Gardens Conference to be held in Melbourne in September this year. The conference theme, Influence and Action, Botanic Gardens as Agents of Change, will explore how botanic gardens can play a greater role in shaping our future. And we've heard lots of great ideas today. With accelerated loss of biodiversity across the globe, increased urbanisation, population growth and climate change, we need to work together to find new solutions for the future. The 7th Global Botanic Gardens Congress will run from Sunday the 25th of September to Thursday the 29th of September this year, and it's being organised by BGANZ, BGCI and the Royal Botanic Gardens Victoria. John's team will be extremely busy. The conference will have a strong focus on plant conservation and restoration, horticulture, innovative public engagement and education, climate resilience, cities and urban habitat, and there's 6 conference themes, field trips, symposia and workshops. If you're keen to find out more about AMPC and BGANZ, the best place to hear about all our events is to subscribe to both newsletters and consider joining today to take advantage of the networking and membership benefits. I'll share the details as we close off today. Thanks again to the AMPC and BGANZ staff and committees for putting together today's program and keeping our networks really active and vibrant. Enjoy the rest of the activities planned for Botanic Gardens Month, which you can find on the BGANZ website. There is a change to this evening's program with a focus on rare plants, large and small, including orchids as a focus. It'll be live streamed on Costa's social media pages, so stay tuned for 7 o'clock tonight. Keep on with the wonderful work that you all do in growing and conserving our national plant treasures. Thanks so much, everyone.